other certificate courses like drug development certification course by UC San Diego University. And SAR also got 25 publications in pharmaceutical sciences and 5 publications in philosophy in various reputed national and international journals. With this small note, I welcome Ravi Kumar sir. Thank you for giving me this opportunity.
So shall I start?
Yes, sir. Sir, you are the co-host, sir. Now,
Yes.
you can share your screen, sir.
Okay. So thank you, Mr. Anil Kumar, for your kind introduction. So before I would like to start my presentation, let me thank to my important thanks first goes to Dr. Dwarakanath Reddy sir, who is my dear friend and colleague in past college. I mean the same college where now you're all working. And Dr. Swarnalata Madam, Principal, ANCP. And all my friends, including Pradeep and Surya Prakash sir, Deepa Madam. So these are all my friends, Praveen sir. So many are there still working after I left the college. And there are many new faculty also joined. I may not knowing all those them, but still my thanks goes to each and every one of ANCP team. So with this, I also would like to thank to management of ANCP for supporting these kind of programs, but not least, I would like to thank to the ACT for sponsoring this program. So with this thanks note, let me proceed to my presentation today. So let me share the presentation. So I'm just facing some technical issue. I don't know. So it's not sharing in tab. So currently I don't have laptop access. So yeah, Pradeep, can you share my?
Sir, can you send the PPT through mail, sir? Let me share.
Yeah. To promote. So I don't know why it's not sharing. Actually, I used to take classes also.
Send your PPT to Pradeep now. He shared it. Don't worry.
Okay, right, sir. Okay. That's what I'm looking for. So Pradeep sir, your number?
Sir, mail ID sir STTP.
Email. So can you tell me the WhatsApp number so that it quickly I can tell you.
Send to the promo sir. Send to the promo. Okay. Already it's connected, sir. Okay.
Please hold on a minute. I'm
no problem.
So, Dr. Pramod, I shared with you. Can you please present that for me?
Yes, sir. 1 second.
Yeah, sure.
Sir Ravi sir, is it
Yeah,
okay now?
now I can see. Yeah, perfect. So, good morning to all the delegates. It's my privilege to present the topic involvement of artificial intelligence as advanced computational tool for drug development. And this occasion of AICTE sponsored STTP. So, I'm working in currently Sri Vishnu College of Pharmacy as an associate professor and head of the Department of Drug Regulatory Affairs. So, next slide, please. So today, objectives of my presentation is to recognize and realize what is the role of artificial intelligence in pharma, and especially in its applications in pharmacy, and show some evidence of AI, artificial intelligence role, especially in pharma industry. Trying to understand a little bit of AI mechanisms, and finally recognize the potential of this area for giving projects to PG scholars. So the one of the major focus my attention is by taking this topic is there is a huge scope to take up projects for the PG scholars, and I hope I am under impression that most of the faculty who are attending this presentation is youngsters. I mean, who are in the early of their career. So that's why some of you may be thinking also to do PhDs. So for this reason, this area, the artificial intelligence and its role in the pharma industry is currently emerging area. So there are a lot of areas, sub areas where you can also think about your PhD work to take up. So these are and also PG scholars projects. So we are currently seeing since almost 5 to 10 years the role of AI in um, IT industry, electronic gadgets, and many other areas. But most of academicians in pharma industry not much aware of the role of AI in pharma, especially pharma industry. And now we are seeing the applications of AI in medical device industry. Almost we know every one of us when we go to hospitals for diagnosis purpose. So we are taking diagnosis by using advanced medical instruments, that is called medical devices. Most of these devices are built in with the AI technology. So that much we know, but most of academicians do not know exact evidences and what is the current applications already started by the AI in pharma drug discovery and drug development. So this is what I feel in recent days. I had got some opportunity to spend some time on this topic, where we have provided by our society a platform called edX program. So uh, the society is offered us at a free of cost, though they are paid courses, but out of courtesy, they have given free of cost those courses to be done for these artificial intelligence, which are designed by IBM. So, currently IBM is on top, taking up the AI as one of the computational tool for the various purposes, various applications, not only healthcare, but various purposes. So they also invented one of the greatest AI platform that is called, you might have already heard about it, Watson. So Watson IBM. It's a big project which is successfully launched in the last 5 years back around. It's doing wonderful job now. So it also coming into healthcare industry, this Watson IBM. So we also come across in the coming slides. So these are some important points I would like to touch upon in this presentation. I request all of you to be patient because it's a dry topic. So the only purpose to be fulfilled. So that's why I have to cover lots of examples. Definitely, it would be somewhat boring, but you can gain some knowledge if you carefully follow the presentation. So yeah, before I start, go to introduction, Dr. Pradeep sir.
చెప్పండి.
So these participants are coming in between and
Yes, sir,
admit,
I'm accepting, sir. No problem. I'm accepting.
Okay, so I don't need to admit, right?
No, yes, sir. Yes,
you're
sir.
admitting.
Yes, sir. I'm admitting.
Okay.
No problem.
Yeah. Thank you, Pradeep. Next slide, please. Yeah. So we are very familiar with the drug discovery and drug development. Almost being a pharma academicians, we know lots of theory and also practicals behind this process. And what is the primary concern associated with the drug design and development is the major factors are time consumption and production cost. So further, apart from these, there are other factors also which are important hurdles like inefficiency, inaccurate target delivery, and inappropriate dosage. So these are also other hurdles for the drug development. Before that, there are challenges and hurdles for the drug discovery facing by the traditional approaches usually employing by the pharmaceutical industries. So these are the drug development stage, the different hurdles are facing by the pharmaceutical industry. So these are the basic concerns and basic challenges facing by the industry. Next slide, please. So in order to minimize these challenges and hurdles, for the past 20 years, researchers are around the globe toward computational approaches such as virtual screening, molecular docking, which are also known as traditional approaches. So this molecular docking we know already, virtual screening also we know. These are also advanced computational approaches. Remember, these are also computational approaches, but these are called as the traditional approaches. But however, these techniques also impose challenges such as inaccuracy and inefficiency. That means they do not have self-sufficiency and eliminate the challenges are their own. So they do not have that capacity. So also accuracy is not possible through these methods. These are the common challenges encountered by the traditional computational approaches. So after this, yeah, next slide, please. Dr. Pramod, next slide. Yeah. So this is, this is emerging methods. Now we are seeing the self inefficiency, inaccuracy of the traditional approaches. Finally, the emerging methods, which includes artificial intelligence, including deep learning, machine learning. I'm going to define and I'm going to give some basics about these terminology in the coming slides. So these has emerged as a possible solution which can overcome the problems and hurdles in the drug design and discovery process. So with the advancements in the technology, especially we know very well the computer-aided drug design, which is integrating artificial intelligence algorithms, can eliminate the challenges and hurdles of traditional drug design and development. So please make note of the point here. This is a kind of combination technology. So we know very well that CADD, CAD technology, so which is almost since 10 to 20 years, which is using in the chemistry drug discovery, especially chemistry part, so computer-aided drug design. So now what is happening is this CADD is also integrating with the AI, so that giving best results. That is not only giving best results, also eliminating the challenges which are facing by the what we call traditional methods. So that is the beauty of the AI technology. So AI is a kind of very versatile technology, very versatile in nature. You can use it in different locations. That's what we are going to see. What are the different areas where AI has its own advantages and applications in the pharma field itself. So different areas. It is not only comes at the first phase. It is going to have its role till product reaches to the market, and after reaching to the market also AI has lots of applications in the clinical phases also. Especially now the role is becoming very high for the AI in the post-marketing surveillance studies also. That's also we're going to see. In the last point, you see the term AI. This is very important. Please make clear the understanding of what you mean by AI here is when we can use the AI applying in the healthcare industry, something like that. So is when machine mix cognitive behavior associated with the human brain. So during learning and problem solving, then we can call the term AI. So what do you mean by artificial intelligence? Whenever a machine mimics cognitive behavior, that means at a cognition level, it's not exactly cognitive behavior like in the human, but comparatively it resembles the human cognition power in the machine. When you see such kind of mechanism taking place in the machine, then you can call it as a AI. So usually you see most of the gadgets, electronic gadgets which we are using, they do not have these kind of capacity. So they cannot mimic such kind of cognitive behavior. But these instruments which are now coming in the label of AI, they have such kind of capacity. They can think at a given situation and they can take up particular challenge and they can give you solution also. So identification and as well as overcoming challenge by giving the solution. These are the roles very effectively performed by the AI, which can mimic kind of human behavior and human attitude. So that is the exact meaning of not definition, meaning of AI when we are using the term AI. So usual ga ee capacity or capability eppudu vaadtamu ante problem solving. So this is the one of the common understanding when we are using the term AI in different industries. So problem solving is the one of the major what we call advantage of AI technology. So next slide please. So let's see, begin with the history of AI in healthcare industry. Of course, AI has its applications in different industry. Now we are not going to bother about those. Well, let us focus only on the healthcare industry. So it begins its journey in the 1950 by development of Turing test. Then it 1971 research resource on computers in medicines, and 75 first NIH sponsored AM workshop. Then 2000 deep learning was introduced. So deep learning is a very important concept in the whole AI platform. Then deep QA technology is introduced in 2007, and 2010 CAD applied endoscopy, and 2015 farm-bot was developed. 2017 first FDA approved cloud-based DL application. This is the one of the biggest milestone achieved by the AI technology in 2017. So then 1820 now there are lots of trials, AI trials are going on in gastroenterology. This is a one example. There are plenty of examples are seen here. The kind of in 2018 and 20, in between 15 to 20, now currently 2021, lots of things are evolving that we will see in the coming slides. So this is a history, begins from 1950 to 2020. Yeah. Next slide, please. So now let's see the classification of AI. So remember, AI is one of the advanced emerging computational tool in the drug discovery, drug development. That is what our topic today, understanding how it is a computational tool for the drug discovery and drug development, and where and what are the evidences, what are the case studies or examples. That is what we are trying to understand. So it has these different classifications in AI. It begins with reasoning and problem solving. So intamundu nenu oka meaning cheppanu. So when we can call AI, so these are the various what we call kind of classification and comes under AI. So if these are all the qualifications, or you can say prerequisites, are there in any system which is established by these what we call software, then we can consider it as a AI. So what are those? One thing is we already discussed reasoning and problem solving, and knowledge representation. That means we give lot of input. So data, big data. So kind of lots of data we will give to the system, but it just not only storing, it also represent the knowledge whenever it is necessary. That is a role of AI. And planning and social intelligence. So that is also advanced. It also mimics the human behavior and attitude. Perception. This is the biggest point, perception. So till now only human beings and living creatures only having this perception, you know very well how we perceive the things. So and current situation. That is only possible actually by the human consciousness. But nowadays, artificial intelligence also entering into this zone. A little bit of perception is possible by the AI. And then machine learning is also biggest concept. We have totally three concepts, two important concepts in the AI, that is deep learning, machine learning. So there is an evolution of this one that we will see in the next slide. Remember that machine learning is also part of AI. And robotics, we know nowadays robotics, lots of surgeries are taking place with the help of robotics, even major surgeries also. They are very costly, but very successful. So that is AI. It's proved already its effectiveness and efficiency in terms of robotics. And natural language processing. So this is the biggest challenge for AI now. When I attended this workshop, they said that it is very difficult to learn the system our language. So that's why the humans are unique always. AI can never beat the humans, because even simple language also understanding by the software, these systems are, it's very difficult process, but it's still under progress. So natural language processing, but it is little far from our applications today. So this is how we can classify the AI. Thank you. Next slide, please. So in healthcare and pharmaceutical industry, this is a very broad areas where the AI has its applications. It has its applications in the manufacturing and supply chain. Actually, it begins from the research and discovery. It ends here in the manufacturing. Sorry, it ends in the consumer marketing. I mean, till post-marketing surveillance. So you take anywhere clinical development. That means the clinical trials, phase 1, phase 2, phase 3 clinical development. There is lot of role nowadays, including recruitment of right patients for the trials. There also, actually, it is the biggest challenge to the pharmacology scientist to select right patients for the clinical trials. But that is now very smartly doing by the AI. And patient surveillance, that is possible. That means we know very well pharmacovigilance is working very hard to identify the side effects and adverse events after a drug is launched in the market. There also now, as I already said, Watson IBM, which is a wonderful AI which is already launched in a few years back, that is doing great job in these kind of areas. So patient surveillance, that means identification of these things. Post-marketing surveillance, we can call it as a pharmacovigilance. There also AI playing very important role. And post-marketing surveillance, it is a phase 4 studies. And manufacturing and supply chain. Yes, of course, there also lots of errors takes place, human errors that can be avoided with the help of AI. So that means if you see these applications, AI applications in pharma industry, almost all areas are covered. So far, this why I'm actually taking up this area is that is the reason why, because although AI is already doing lots of jobs in the pharma industry, still most of us academicians, even me also, till I attended those courses, I also do not realize that AI is already entered into pharma field in various zones and doing lots of jobs. So it is important for us at least now to realize and recognize the importance of these AI and to explore the concepts and at least start our journey in the AI, because the coming era and future era depends on these kind of technology based development. So that's one of the important reason for me to take interest on this topic. Next slide, please. So now evolution. So here I just would like to remind all the delegates one point, that if you anywhere stuck in between, you please raise your hand, or you can interact with me in between also, because the topic is very dry. If I go in monotonous way, that means one way, you may feel bored, and if you don't understand or if you not follow my pointings anywhere, you can please stop me and interact with me. So yeah, now coming to the evolution. So evolution of AI, it begins from machine learning to deep learning. So as I already told, AI is not overnight success. It started its journey long back, but especially it has three components in it. The two components are part of AI. Overall, the three components. So some describe ML, machine learning, as a primary AI application, while others describe it as a subset of AI. So that means it is still the debate is there. we keep it aside. If you take in machine learning data are inputted in the machine along with an algorithms. We know about this is all based upon the algorithms. All these deep learning, machine learning, AI is all worked only on the algorithms. So that's why if you want to do job in the AI industry, you must be qualified person at least in some mathematics and statistics. These are the two important prerequisites for you to work in the AI industry. So you must know at least how to give the data in terms of algorithms, how they work. So some basics you should have. You may not be an expert in those areas, but you should have at least some prerequisite qualifications. So the first time in 20th century, Igor Eisenberg and his colleagues, first time they used the term called deep learning. So this deep learning term they introduced in 20th century. From that onwards, this evolution took place. Then machine learning and today artificial intelligence. But remember, whenever we use AI, AI is a not separate segment apart from the deep learning and machine learning. They are the parts and components of AI. So this is to be understood. Yeah, next slide, please. So these what I am trying to here giving the definitions, understanding the terminology. Please be attentive. Otherwise, if you don't understand the meanings and mechanisms of this AI, then it would be difficult for us to understand the applications also. So how the machine learning uses either of anything. So we are calling it as a machine learning. How a machine is learning. So it is on different processes. So they are supervised learning, unsupervised learning, semi-supervised learning, self-supervised learning. So supervised learning means the model is trying to use the labeled data, means inputs has been tagged with the corresponding preferred output labels. So it's something, it's not so typical to understand, very simple, that we have already accumulated some data with us through some research. We have done lots of research. Our scientists have been doing research since some decades. So all the data is accumulated now. What we do is we give all the data to the system and we label it. So that is called label data. We label it, we tag it. So this is the data of this particular molecule, something like that. We are naming or we are labeling the data. That is called supervised learning. Unsupervised learning, here the model is trying to use unlabeled data. Data cannot be given here. Unlabeled data will give, and there are certain algorithms and some patterns are there. So based upon the structure, based upon the size, different categories, based upon different parameters, the system has to segregate the data into different clusters. So based upon that, it is going to learn something. Okay, these molecules are, if they are in this size, they are in this pattern, then they comes under this category. So something like that, it's going to use or analyze the data based upon different patterns. Whereas in the first supervised, the label is already given to that. Based upon the label, the data, whatever given, is going to segregate it. So that is called supervised, and this is unsupervised learning. Semi-supervised, nothing but combination of both. Some labeled data is given, some unlabeled data is given. That means we are training the machine in a different ways. That is all the biggest job doing by the AI scientist. AI scientist chesedi em ledhu ikkada. So first they have to give continuously training the machine that, so different conditions, different situations come, then the system has to have a capacity to take the decisions on spot. So how that is possible? Only by giving proper training. So the training can be done in these ways: supervised, unsupervised, semi-supervised, and self-supervised. So self-supervised, special cases, two-step process, ultimate goal, make supervised learning. So first two are more important. Remember, supervised and unsupervised learning. Mostly these two processes are commonly using by AI in machine learning processes. Next slide, please. So in deep learning, what happens? Deep learning, many layers of machine learning algorithms, which is called brain-inspired family of algorithms, which mimics the human. This is very important. Please understand. Very simple terminology. What do you mean by deep learning? Already machine learning is kind of algorithms. It's all based upon the algorithms. We have a different data. We give inputs. All the data given to the system based upon the algorithms. Machine is going to learn based on the algorithms. But here, what's the difference between machine learning and deep learning? Deep learning is a kind of many layers of machine learning, and already lots of layers of algorithms are there in the machine learning. If you put together all of them, then they are going to become as a deep learning. So different models, combination of different algorithms in the machine learning, then it goes to the next level of learning process that is called deep learning. Ante. So for example, if you want to understand in simple ways, there's lots of processing taking place in the human body. So right now, so but where the more complex learning or complex network is there comparatively, it is in the brain. So because there is lots of neural network is there in the brain, lots of processing also taking place compared to other systems in the body. In the neural network I'm talking about. So similarly, so in the machine learning also lots of algorithms are there, but in the deep learning what happens, it is almost like a brain inspired family of algorithms. So that is why deep learning mimics the human brain. That is the meaning of mimics the human brain. There, these complex algorithms, due to the complex algorithms, the deep learning, it kind of resembling the human brain. So that is how it is labeled as a artificial intelligence. So, but remember, this process is not as easy to the AI scientist to make into that deeper learning process to the system. To help the deep learning system, high computational power is required. Lots of training and big data is there required. The big data I'm going to define in the next few slides. So this is the difference between the meaning of deep learning and machine learning. So here the deep learning is still in the growth phase, and creative ideas are required for the further advancements in the field. As I already said that it is very complex process. If you want really mimic the human brain, it's not so easy, because even today doctors also, especially this neurologist, also not able to understand certain things in the surgeries, neural surgery. That's why you might be knowing that if you want to perform the surgery on the brain or something related to this neural network, it takes very long hours. Nearly sometimes they take 8 hours, 12 hours also for performing one simple surgery. So that shows the complexity of this neural network. And so that is why if you want to build a system by mimicking the human brain, definitely the process also very hard, but still they are making good remarks, the good progress in this area. Now coming to a very important concept that is called big data. So after, if you really want to understand the applications of these AI, DL and ML in the any field, you must know at least some basic idea about, some basic information about big data. So what is this big data? Big data can be defined as the data sets that are too gigantic and intricate to be analyzed with the conventional data, analyzing softwares, software tools and techniques. That means very massive data already available, which is so far based upon the researchers performed. You can find the data from the different literature, some different sources are there in the internet. So you can collect so much of information in any given field. So like that we also have in pharma field. So that is called massive data. And the data is how it is, it's very complex. When you have a lot, so much amount of data, usually that is complex. So that data to be analyzed with the conventional data analyzing software tools and techniques, that is called big data. That means we are pooling all the data and finally analyzing with the software tools and techniques which are already available, conventional analyzing tools though. So that process is called big data. So what is that? Simple two terms: collection of massive data which is already complex, then analyzing with the conventional tools which include software tools and techniques is called, put together all these processes and procedures, it is called big data. So I hope you got the meaning of this term big data. Yeah, next slide please. Yeah. So there is a big role of big data in artificial intelligence in the drug discovery process. We are going to, from here, we're going to have lots of examples in the coming slides. Of course, it is, though it is dry and boring, but still we need to realize, yes, there is lots of evidences are already there. It is not about only theory, talking about theory, but practically seeing there are no much significant improvement or progress is there. It's not like that. There are already ample of evidences are available in the field of AI and big data. So actually you need to remember 3 points when you're talking about big data, that is volume, velocity and variety. So based on these 3 concepts, all the big data is going to work. So what are those 3? Volume, velocity and variety. So volume means, you know very well, it is a quantity. So there is already we have taken the meaning that big data means it is a huge volume, massive data. So gigantic data. And velocity means the process of analyzing, how much time it takes to analyze the data. And variety, what are the difference in the what we call pools of data. So although we collect the data from one particular field, but still you can see lots of variations in the data. It is never possible that all the data which you collected must have a similarity. Usually lots of dissimilarity you can find in the data which you already collected. That is called variety. So these three important features the big data usually having, and based upon that only it works. So here you see arrival of microarray, RNA sequencing and high throughput screening HTS technologies helps to produce large amount of biomedical data every day. Thus, drug discovery has made a transition into big data era. So usually we'll get a doubt here. So is it possible to pharma industry to have that much data to process by using these kind of tools, advanced computational tools? Yes, the answer is very simple, that these after arrival of microarray, RNA sequencing and HTS technologies, high throughput screening technologies, they are the biggest sources for the pharma industry to give large data on day-to-day basis. They are producing lots of data during the research in the industry. So that data is using as one of the biggest source for the big data analysis. So that is why the drug discovery nowadays made a transition into the big data era, from that drug discovery big data era. So that is possible only by these technologies, because they're able to give the lots of data inputs. So I hope you got this point. Next slide, please. Dr. Pramod, are you there?
Sir?
Next slide, please.
Sir, one second, sir. One second. Sir, is it okay, sir, now?
Yeah, now I see the change in the slide. So is there any problem in changing slide?
Sir, actually, showing some kind of antivirus updates. That's why.
Okay. So I'm sorry for troubling. Actually, I should suppose.
No problem.
So let's continue. So I think, yeah, it is next slide. So the target identification gene expression also helps widely. So we see the big data and AI applications, clear evidences here, and some important areas also we're going to cover here. The target identification, you know very well that how important it is at the beginning of research in the pharma industry, target identification. So that is gene expression, target identification, gene expression is the one area. It also helps very widely for the big data. So for example, big repositories, big sources of data containing gene expression data, NCBI gene, the cancer genomic atlas. These are the different sources, platforms where you will find lots of data related to these particular fields. So that is where all the data is pooled for applying the big data analytics. So example is given here, machine learning approach and gene expression data. So here this is a scientist called Van Ijnondor in 2019 found out novel biomarkers and potential drug targets for the rare soft tissue sarcoma. How did he achieve this particular novel biomarker? By collecting the data from the big repositories given in the above paragraph, that is NCBI, GEO and TCGA. By collecting all the data and using big analytics, big data analytics, he found out these novel biomarkers which are helping in the soft tissue sarcoma. This is how the machine learning process can help. So what he done actually, ML approach and gene expression data. So he combined together and he found this novel biomarkers. This is one good example. We understand how it helps the machine learning process. Next slide, please. And the databases and repositories, you find here, I have given some examples of databases and repositories, PubChem, ChemBL. There's lots of data available on different specific fields. You can find the specific fields of information. This is just example I given databases, because big data is largely depends upon the data only. So you need to have a lot of data for processing the this one. So that is coming from these other different sources of information, that is databases and description. I don't need to read out all those things, but just for information sake I kept here. Next slide. Yeah. So here you see, after discussing all those things, we find here what are the different applications of big data for drug designing and discovery. So it actually starts its journey right from chemical compound database first. Chemical molecules, I mean molecular information. There are some platforms are there, ChemSpider, PubChem, etc. From there they'll get all the information. Then it moves on to physicochemical properties or data, pharmacological data. There again you see lots of data available on the physicochemical properties. That data also can take and process it by using big data analytics, and then by applying machine learning models. Up there itself filtering of the compounds takes place. In the third image you can see between filtering of compounds, there also there are some techniques the AI techniques are using to filtering the compounds. Then machine learning models, and comes to the this classification, hierarchy of sorting, etc. These are all chemistry part. So the terminologies we may not know, but at least we know the process. Then QSAR, it is also part of chemistry research and pharmacophore modeling. There also the applications of this AI is there. Then it moves to the protein and ligand structure. These also we see lots of examples in the coming slides, where nowadays AI is having significant impact on these protein binding, receptor binding mechanisms. Then visual inspection and compound selection, experimental validation, and finally in vitro and in vivo experiments, including clinical trials, drug protein interactions, drug bioavailability, polypharmacology, drug monitoring, drug dosage, etc. So almost right from beginning to till last in vivo studies, every place, everywhere, the big data and as well as AI is having applications. So the examples we will see in the next slides. These are the just basic flow chart where applications are possible for this field in the pharma. Yeah, next slide, please. So applications of multi-scale methods in drug discovery. So I'll just give a few examples here. One is molecular dynamics of cardiac modeling. So cardiac modeling is one of the big area for the pharmacy research, where lots of diseases are located around the this heart, cardiovascular diseases. There definitely the lots of exploration is taking place in this field, that how AI or machine learning can help to find the suitable models and also to bring successful molecules with the easy efforts. That's why lots of research is already in progress on this area, cardiac modeling. So here structure modeling of ion channel interactions with the drugs is critical approach for current and future drug discovery efforts. What is that channel interactions, ion channel interactions? As we know already, if you're a pharmacologist, you know better how the ion channels are playing very important role in the heart functioning. Whenever there is some small defects takes place in the ion channels, also immediately it affects the flux of the either potassium or calcium or sodium, these ions, that leads to blood pressures and different diseases. So here the structure modeling of this ion channel actually in the process of drug discovery is a very important approach. So for current and future drug discovery efforts, here in the modeling of drug receptor sites within an ion channel structure can be useful to identify key drug channel interaction sites. So modeling of drug receptor sites, you have to model such a drug receptor site within an ion channel. So for example, you take a sodium potassium pump, within that channel you have to make such a receptor, so to identify key drug channel interaction sites. So here structure modeling of drug channel interactions at the atomic scale may ultimately allow for the design of novel high affinity subtype selective drugs. As we know that, for example, you take a H1 blockers or H2 blockers. So there are some drugs are there which are working for these BP and etc. So these drugs are mainly functioning or working based upon the affinity, receptor based, how strong they are having affinity to bind with the receptor sites, especially related to these ion channel blockers, etc. So that high affinity molecules are possible to develop based upon these kind of advanced modeling technologies. So where AI and these deep machine learning, especially machine learning, having some significant role playing. So that is called successful developing of these models for bringing of novel high affinity molecules, especially cardiac and neurological disorders, because these are very complex areas in the healthcare industry. Next slide, please. So already lots of what we call work is going on in the Alzheimer's disease and epilepsy using these kind of approaches with the help of machine learning and AI. ANN is a one classical example for that machine learning techniques that is already proved its efficiency in the clinical analysis of big data, which is related to this area of working, that is Alzheimer's and epilepsy area. And Han's model also, Hanna's model also is a classic method of solving quantitative structural binding relationship in pharmacology and medicinal chemistry. This is also similar kind of model, which is helping in the establishing of the advanced model for bringing out successful and novel and more affinity molecules. So there's another model called PT and QSBR, combination of these methods, PT QSBR model, which is for a large number of drugs reported in Campbell, focusing on a protein expressed in the hippocampus of the brain Alzheimer's disease patients. That means they also tested one model on the Alzheimer's disease patients. They got some positive evidence of working better of these advanced machine learning based models for the developing or working seriously on this area with the help of this AI technology. These are all the evidences I gathered from the literature in order to show you that the significance, impact, and role of the AI and machine learning in terms of drug discovery and drug development process. Yeah. Next slide, please. So, congregation of artificial intelligence and conventional chemistry improves drug discovery. So, which is also that means taking the help of conventional chemistry and applying the technology of AI to bring some advanced things, that is advanced models. That is also one of the one of the good approach following by the pharma industries nowadays. That is AI algorithms such as ML, DL, machine learning to deep learning have been increased in CADD. Earlier CADD alone is working, now it is combined with the ML and DL. So these whenever you talk about this, AI techniques provides answer for two important questions in the medicinal chemistry, such as what should be the next compound, what is the process of making a compound. Because what is the next compound is a very difficult question to answer by chemistry during the research. That's the biggest challenge for him, because you are going to actually screen hundreds and thousands of molecules. So that's why it is very difficult to predict based on the conventional methods. And also process, you know how difficult it is to scheme the process for any one compound, or through which process, which easy process, which simple process, which economic process you are going to make that compound. That is also biggest challenge every time to the scientist. So in these two areas, AI has found some important applications. Yeah, that is possible by the congregation of these two. AI alone nothing can do. Remember, I would like to make a remark here. AI alone can do nothing in the healthcare industry. Everywhere it required the support of the conventional and traditional methods. That means a human effort. Human effort is needed, and human produced data is needed by all the traditional approaches, by taking the help of the data and the skills and the knowledge of the human effort on the different fields, maybe chemistry, pharmacology. Then only you can take the help of the AI to bring advanced models, computational models. So AI alone can do nothing here. Yeah, next slide, please. So these are the examples to distinguish the compound cell activity done using machine intelligence methods such as DT random test method, CNN LSST network, and gradient boosting machine. These are also I brought to show some evidence here. Examples: classical ML machine learning methods and DL methods could classify the compound cell activity. So that means it is a proof that they are already successfully classifying the compounds cell activity. And AI and MA also helps in distinguishing existing drugs and novel drugs. That is also a beautiful thing. If you have a big data, then you can possible to segregate what are the existing drugs and what are the novel drugs. So, for example, Zhang et al. Implemented computational analysis against novel coronavirus, where the authors screened different compounds that were biologically active against SARS disease. That means these advanced computational tools are already using for the coronavirus also. There's some positive outcomes are already reported in the literature. Next slide, please. So transforming traditional computational drug design through artificial intelligence and machine learning techniques. Transforming traditional, that means we are moving ahead by taking the traditional approaches to the next level with the help of AI. So for example, Google DeepMind has advised an AI-based tool training on PDB structural data referred to as AlphaFold, which can predict the 3D structure of proteins from their amino acid sequences. That's the advanced approach. It's a kind of advanced model. It's not so easy to predict the 3D structure of the proteins. It's a very molecular level kind of mechanism. So without these kind of advanced tools, it is very difficult. With a highly sophisticated and costliest equipment also, successfully one cannot predict the 3D structure of the proteins. But it is possible. Nowadays, it is also proved in the Harvard Medical School. A team of people approved a DL-based tool that takes protein's amino acid sequence as an input and generates its three-dimensional structure. That is one big thing happened recently. What they have done is they have taken the amino acid sequence as an input and generated three-dimensional structure. So that means you are able to predict the three-dimensional structure of the protein by this experiment. That experiment has done successfully in the Harvard Medical School recently. So this is how it's moving ahead. Yeah. Next slide, please. So this another example, SNORB DL driven tool can help researchers in designing new pharmaceutical drugs, and AI has the capacity to accelerate the molecular dynamics simulation process. And de novo drug design has also taken the advantage of AI. So these are all the various areas where in the drug discovery, especially at the beginning level, AI is taking what we call is providing support to the scientist. The example is given here. 2020, Q Boy et al. Is reported a work that is a tool that can design three-dimensional drug in three-dimensional protein pockets. So that is also reported in the literature. I collected it. Then text mining tools of AI such as NLP helps to transform the unstructured data to structured data. So this is also important. We have lots of data. Some of them is structured, some of them is unstructured. Whenever you have a structured data, it is easy for you to move forward in the research. When you have unstructured data, which is not categorized data, then it is very difficult. It is almost, you can say, it's not useful at all for you to do the research with the data. So that is why you need to make it into unstructured data to structured data. In this process, AI is having a lot of help. In that, again, there are a lot of subfields, sub models are already established in which are based on the deep learning and machine learning, which are helping to process this unstructured data to structured data. Next slide, please. So this is again primary and secondary drug screening, the applications. So the primary drug screening includes the classification and sorting of cells by image analysis through AI technology. So here you see usually primary drug screening process includes this classification sorting. What are the commonly using technologies in the AI is image analysis through AI technology. It's one recently what we call investigated method, which is successfully implemented also in many industries. And ML models, that means machine learning model, needs to be trained so that it can identify the cell and its features done by contrasting the image of the targeted cells, which separates it from the background. So how that works, that is what the mechanism is given here. It is trained, machine is trained in such a way that it can identify the cell and its features, and it can even separate based upon these features. So compounds can be separated based upon these features, which are already trained by the humans to the machines. So for example, how it works, you can see in the last last point, that is the study suggested that least square LSSVM method. It shows that highest classification accuracy 95.34% is achieved by using AI technology. That means you are able to successfully segregate, and the compound screening is done, made possible up to this extent. 95.34% is not a small thing to be achieved with the help of a machine. That means technology based. That's one evidence we see here in the primary drug screening process, the application of AI. The next slide, please. In the secondary screening, analyzing drugs physical properties, as we know that once you screen the compounds, you need to analyze its physical properties. There also there are some models are established. One example is matched molecular pair, which has been extensively used for the QSAR studies. There also you find lots of applications. I don't need to go on telling that molecular fingerprinting methods, simplified molecular inputs. This SMILES model is very popular in terms of secondary screening applications by the AI. So one can find these all already reported evidences and articles also find in the literature. So that means finally we can say that in the primary drug screening and the secondary drug screening, both places AI has its significant applications. Next slide, please. So I don't take much time to explain this one. Just want you to show that here you take this advanced computational tool, that is AI and big data, ML and DL, having their applications right from lead identification to primary screening to lead optimization, lead compound identification, secondary screening, preclinical and clinical development. And everywhere, as in the first slide we discussed, the major concerns about our traditional research is what is that? Is the time consumption. They take a lot of time to complete this process, and as well as cost of the production. So both can be reduced and minimized with the help of artificial intelligence and drug molecular determination approaches, that is machine learning also supervised, deep learning, unsupervised reinforcement learning. All these applications by put together, they are able to produce all these complete these steps hardly in 4 to 5 years. That means this technology is made possible to reduce the time from 10 to 5 years. That means almost 50% of the time is saved in terms of research. And of course, whenever you are reducing that extent of the time for any purpose, you can understand how much money is saved. So that means cost of production usually comes down drastically. So that's one important point I would like to what we call tell here at this slide. I don't want to take much time here. Yeah, next slide, please. So now we're coming to the final phase of the presentation, the direct applications of artificial intelligence in the drug development process. So there are lots of applications already made possible and what we call shown evidence also in the industries, which are also reported in the different literature. So a few examples, important examples you can see here. One is peptide synthesis and the small molecule design. So there are examples given here. DL based platform, N et al. In 2020, they have established model called Deep AMPEP30. So this is a platform for the identification of short antimicrobial peptides. And another scientist, Plisson et al., the group of people combined this ML algorithm with an outlier detection technique to discover AMPs with a non-homolytic profiles. So like this, you can find lots of examples here in the peptide synthesis and small molecule design. This is one area of application where in the drug development AI is found. Next slide, please. Then identification of drug dosage and drug delivery effectiveness. So here also, this is also another important area where drug dosage and drug delivery effectiveness, because once you developed a molecule successfully, the next immediate question to the pharmaceutical scientist, a formulation scientist especially, to find out which dosage form will be effective for this particular molecule. That would be a challenge for the formulation scientist to go for trial and error basis. Lots of dosage forms they have to test, whether tablet, capsule, powder or liquid or nano formulation. The different approaches are there. That also takes lots of efforts. So there also AI has really now emerged to give you some some help and support to find what is a better dosage form, effective dosage form for you to fulfill the purpose. So here, the one example I reported here gathered from here to from the literature, that is Shen et al. Administered a combination of tenofovir and efavirenz and lamivudine to 10 HIV patients. And in due course, using the PRS method, they found out the dose of tenofovir could be reduced by 33% of the starting dose without causing virus replace. This is a significant, what we call significant contribution done by the tool, this AI tool, in finding the exact what we call dose appropriation, that is 33% of starting dose. So this is a kind of finding effectiveness of that particular drug in the drug development process at the later stage of drug development. So another application, another example also here mentioned, using combo FM, identified a novel combination of anti-cancer drugs, crizotinib and bortezomib, showing promising efficacy in lymphoma cell lines. Lymphoma cell lines are cancer studies. They found some kind of significant what we call achievement in terms of using AI in this drug development and finding effectiveness with the help of AI and ML. Next slide, please. Then structure-based and ligand-based virtual screening. These are all very popular, LBVS and SBVS. Without these structure-based and ligand-based virtual screening, it would be difficult for you to go into the binding studies. So this is also kind of drug development studies. These are the examples I collected. I don't need to explain these examples, but you can understand, oh yes, there is lots of applications with the AI here in this area also. Yeah, these are the examples later, if you can just go through it. Next slide, please. These are the different examples reported in the literature, applications of AI algorithms, including machine learning and deep learning principles in structure and ligand-based virtual screening. You see lots of examples already reported. These are all the very recent and latest information I collected just to show you some evidence of different applications. So just to convince you that yes, this is a very emerging area and one can look into and one can do lots of work in this area. To prove that, I just gathered all this information. Next slide, please. QSAR modeling. So actually QSAR modeling is a one area where I already explained you that how the AI helps to combine with the, in combination with the QSAR to work effectively to establish and to give best results. So actually it takes place in 4 stages: data set compilation and data set processing, model construction, and model evaluation. This is how it goes in 4 stages. But in the interest of time, I cannot explain all these deep flow chart. So at least you remember 4 stages, how the QSAR workflow along with the help of AI. Yeah. Next slide, please. And drug repurposing. This is nowadays you see in the one of the important what we call regulatory strategy following by the so many companies, pharma companies, to making easy money. That means to come into the market. Nowadays, of course, you know how difficult it is to find a completely new molecule, to file a NDA application and to get successfully approved a molecule. That is almost, you see very few, 1 or 2 compounds from the thousands of pharmaceutical companies from India. And coming to what is then where they are surviving, we know very well that generic market. India is dominating in the generic market. But now, even in that market also, lots of competition is going on between the companies. So that's why finally drug repurposing is the one regulatory strategy for so many companies, where you already take the existing drug and find the new indications. So that is how you approach. There also, you are going to use AI technology to find some easy and advanced tools to give best results. Say, for example, how it proceeds. Data collection and data processing. First, you do all the data, you collect it and you process it with the help of big data. Right there, you have application of big data, and then it goes to the computational model generation. These are the very common computational model generation, and that is logistic regression, random forest, deep learning, matrix factorization, etc. These are the common computational model generation in the second stage. Then You're going to generate the proof concept, then evaluation for the repositioning methods, and finally validation of repurposed drug. This is how the flow chart usually takes place for the drug repurposing. Almost in these every stage, AI is helping the companies to find and to easily get successfully complete the repurpose drug to brought into the market. So for example, yeah, that's fine. 12 promising drugs recently in 2020 identified for the for the coronavirus. So that is a one good example. We can understand how these helping in the current and latest challenging diseases also. That is possible by DL approach. So here, this is just I made a list of different areas, right from small molecule design to identification of drug dosage and also effectiveness, prediction of bioactive agents. All these are the areas which we discussed so far, where AI is involved in almost every stage of the drug development process. Once again, I'm just making clear that these are the different areas covered already in the AI. Next slide, please. So possible role in the manufacturing and clinical trial design. In the interest of time, I think I'm already exceeds the time, but I'll just very quickly go through coming slides. Hardly it take not more than 5 to 7 minutes. So this is where IBM Watson, I told you already, it's already in the healthcare industry, which is already very popular. You can just Google it, IBM Watson, you will find it. It's a remarkable achievement of the IBM company. They launched this. It can speak, it can talk to you, it can debate you, it can you can perform in competition, quiz competition with you, but you can't beat the IBM Watson. So that means that powerfully made, very intelligent AI. So that's, it's not exactly robot. You cannot call it as a robot, but it is a it's a AI. So you can call it's the best demonstration of AI. So that is also IBM is launched this Watson in the healthcare industry, finding lots of applications of this one. So here is example given that they're using for the clinical trial data. So you take all the clinical trial data available so far in the records, that finding finally create a detailed clinical finding profiles. So because see, pharmacovigilance, if you see the post-marketing surveillance, what the job is all doing about collection of all the profiles and finding out what are the adverse effects are at the different doses and making profiles. Almost all the pharmacy students also doing routine job in the hospitals in the clinical as a clinical pharmacist is the same that. So here the AI, you can do better job if you have a sufficient data with you. So all the data you can put it here, and then finally you can make a good suitable profiles according to requirement. You can make categorize that information, and you can find different clinical finding profiles also. That profiles will help you a lot to understand the side effects, adverse events of the drugs after marketing. I mean, in the phase 4 stages studies. So AI models can also reduce the cost of clinical trials by enhancing the success rate. This is possible. Clinical trials are very costly, as we know that, time-taking process also, and especially if trial is failed, then you know how much it is burdensome to the company, and also economically it affects the company growth. But here, if you use the AI technologies, definitely there are chances of success will bring to the higher level, and as well as time also saves a lot. So that is one of the application. And example, the outcome of phase 1 and phase 2 trials predicted was based on the DL and calculated probability of possible side effects and pathways activation score, which was further used to train the model. So what is this here? That means the data which is already have the company, which processed by the AI or DL models, and it will tell you what is the possibility that you're going to success after phase 1, phase 2 studies. That means based upon that you can proceed, whether rather blindly proceeding without any such kind of hint for the success. It is always better to have such kind of prediction, then you can invest. So that is what AI can contribute to the companies in order to give you the chances of success, higher rate of chances of success. And there is a coming project, visual physiological human, which is made useful for the in silico trials in coming future. Is also, it's not yet launched, but it is another project which is under progress. Yeah. Next slide, please. So there is neurodegenerative diseases. We know how much success the scientists of pharmacy and medical field to find to get a better molecules for the neurodegenerative diseases like Alzheimer's disease. There are many other diseases you see. You find very little amount of drugs and effective drugs on these cases, neurodegenerative disorders. Now the scientist or the healthcare industry is after the artificial intelligence to find some good approaches and good possibilities to get success in the field of neurodegenerative diseases. And there are some, I searched for some evidences to, is there any significant contribution done by the AI in this field? Yes, already some things are reported. One example I brought up here is telmisartan was proposed as a potential repurposed drug for AD, Alzheimer's disease, by using a genetic network driven classification model. So like this is actually how they have done is based upon the enzyme mechanisms. They have kind of ML techniques are also being used to find a suitable inhibitors of target proteins implicated in the neurodegenerative disorders. Based upon that, maybe they take some existing drugs and to find the efficiency or a new indication, a kind of new mechanism. Through new mechanism, it can work for neurodegenerative diseases. That is why, so already launched drug telmisartan, now they found a new indication which can also be used for the neurodegenerative diseases with the help of the models, which is of DL model and ML models. So there is another example also given, but that is enough. Yeah, next slide. So these videos links also there, but I do not have a time to play for you. Please don't click on the video. It takes a lot of time because I'm very interested to show you IBM Watson for oncology, how it is working. Now later you can find out. Manipal hospitals already demonstrated how successfully they are using IBM Watson for oncology studies, cancer treatment in India. But we don't have time now to play that video. But you can later see that. You can also directly search in the YouTube, you can find it, this video. Very interesting and lots of insights you can find. Yeah, next slide, please. Of course, future challenges is advances in AI algorithms, especially in DL approaches, along with improving architecture hardware and easy accessibility of big data are all indicating toward the third wave of AI. That means there is a lot of growth actually indicating for the AI, especially in the deep learning approaches. As we already discussed in the beginning slides, that deep learning is still growing area because it is a very complex process. It's a kind of mimicking the human brain. So there is a lot of work to be done by the computer scientist in the DL processes, where of course it is yet to carry out a lot of journey. So that is what you see. Nearly now 230 startups are there by June 2020. This all indicating that lots of kind of new innovative hubs are coming for this, working on this area. So, but despite great success, this AI is facing a lot of challenges also. That is especially acquisition of data. See, remember one important point I would like to make here: without data, you cannot do much things in this field. Especially when you want to use a big data science or AI, etc. All these AI, ML, DL, and big data science, whatever you want to work successfully, you need to have a data. Without the data, you can't do miracles. So, but not like other industries. If you want, for example, you take any other industry like computer industry or electronic industry, where it is not that much difficult for them to pull the data or at least to find the data. But in case of pharma industry, you have very minute data for the drug discovery and drug research. So that is why nowadays one of the important initiatives taken by the international platforms, international organizations, they established websites and requested all the companies to put all the data, whatever generated by their R&Ds, into the one platform, so that the data can be used by many companies to establish advanced models. So please understand this point. In order to establish advanced models, you need to have a big data. Big data means gigantic data. You need to have a kind of massive data. That data is not available in the pharma field. That is one of the biggest challenges. So that is also now addressing by so many solutions by different companies. So next thing, please. Next slide. So these are already I've told you that possible solution. So they have started some initiatives and asking companies to put the data on the sharing platforms. And apart from that, molecular representation is also a challenge, as it is one of the governing factors in model building. And pharmaceutical companies faces trouble recruiting sufficient number of patients for clinical trials, where also this AI is helping. AI approaches will help identify and recruit target patients, will also help in managing the collecting data. I also covered this point in the earlier slides. Next slide, please. And patenting, vigorous copyright work, and security are also major concerns for AI. In Kakunda, are there any challenges in this field? Means the biggest other challenge is this IPR, intellectual property rights. Yes, there are lots of challenges are there, lots of hurdles are there to move forward and apply these concepts in the realistic world, that is called IPR. So we do not go into the details of those, but IPR challenges are very much there for AI. And finally, faster computation will be required for handling big data. And it is said that in future, the current supercomputers will be replaced by the quantum computers or another technology which will do the job in minutes rather than taking hours. That means they're expecting that in very less amount of time, the huge data can be processed very easily by the supercomputers. Because supercomputers means kind of AI. So it is very difficult for a novel drug target developed by AI to enter into the market even after a great success story also. That is a big thing. It can help and support in the process of drug discovery, but it is quite difficult for AI to bring completely one new molecule based on totally AI models. That is the biggest challenge. But recently certain thing happened, which is first time ever happened, a novel target and its novel inhibitor has been proposed through AI-based tools. That is, it is yet to come into the market. If it comes, then it is a great remark and great milestone achieved by the AI platform. Next slide, please. So this molecule has shown good efficiency in the human cells. Already some trials are going on. In 2020 December, in silico nominated the small molecules inhibitors for IND application they've applied. They are going to start clinical trials. If that comes successfully, then it will be the first time ever where a novel target and its inhibitor was proposed through. So that's the one. Yes, you have to wait for this result. We have to wait for this. Next slide, please. So I would conclude the topic. Though there are some unavoidable obstacles and tremendous amount of work has to be done to incorporate AI tools in the drug discovery cycle, there is no doubt that in the near future, AI will bring revolutionary changes in the drug discovery and development process. So my agenda of the presentation is to bring some awareness in the youngsters, especially to find the good areas in this platform, in this computational tool, advanced computational tool for your research work, and to give some projects to PG scholars to explore more, because it is going to be have a big impact in the next 5 to 10 years. So that's why I brought all these topics to in this presentation with lots of evidences also. I brought only to convince you that yes, this is a very potential area to take up work and to do lots of publications also. So I request you can do lots of wonderful publications in this area. So you can segregate into different areas. You can just collect lots of information. You can do review articles and research articles also. Yeah, that's all. Thank you. Thank you everybody for your patient hearing.
Thank you, Raghunath sir. Your presentation has been enlightening, and your presentation was very clear and was very informative. And this clear information and effectiveness reflected on the number of participants, where I have seen from the starting to the end of the presentation. So the participants were continuously increasing, and has shown more interest on your explanation part regarding the particular topic, sir. So thank you for accepting the invitation for delivering the talk in this phase 3 of STTP program sponsored by AICT, sir. Thank you once again for giving a very clear and effective information on the specified topic to our delegates, which has gained a better knowledge. Especially, you have been mentioned for the PhD and PhD scholars, which is going to be useful topic. So thank you once again for delivering the talk in our STTP, sir. So Pradeep sir,
1 second, sir. So 1 second, sir. 1 second. Sir, 1 second, sir. Sharing.
Yeah.
Yeah.
Yeah, before we go, I would like to thank you to Praveen sir. Thank you for your feedback. I'm so happy, and it's my pleasure to spare time for ANCP always, because
Yes,
yeah, as my friends always there. So especially Dwaraka sir and you and many are there. I'm so happy to always spare time for ANCP, because I came
as
from
if-
there. I never forget that. Thank you.
Yeah, sir.
Yeah.
Sir, as a token of appreciation from Anamacharya Kala Pharmacy, a virtual e-certificate has been displayed, sir, and the same certificate will be mailed immediately after the session, sir. So kindly accept the certificate, and thanks for delivering a beautiful and wonderful topic for our delegates, sir.
Thank you so much. Thank you so much. I'm really happy and honored,
Yes,
feel
sir.
honored to be part of this program. I'm
Yeah,
really
the
thanks
participants
for inviting me for this.
and the participants were also been very stable from the starting to the end of the session, sir. Okay,
Thank
sir.
you.
Thank you, sir.
Thank you. So shall I sign off? Sign off, or any questions are there from audience or anything like that?
Chat box is full of appreciation. So
Appreciation, sir. There are no questions,
Yeah.
but it indicates that how effective the presentation were being delivered by you, sir.
Yeah. Thank you.
Okay,
Thank
you.
you. Thank you. So I thank you all the organizers of the event, especially Dwaraka sir and Principal Swarlata madam, and all the organizers of the event, including Pradeep sir, Surya Prakash sir, and everything, Deepa madam, all my friends and colleagues. If I forget any names, please excuse me. So you're all in my hearts. Thank you so much for this opportunity. Thank you, sir. I'm signing off. Right?
Yeah, sir. Pradeep, sir,
sir.
can I start, sir?
1 second, sir. 1 second. Yes, sir. Yes, sir. You can join, sir. You start, sir.
Yeah, good morning. Once again, I welcome the delegates to the 5th day, second session of AICT sponsored e-STTP program. I, Mr. M. Praveen Kumar, Associate Professor at Department of Pharmaceutics, Anamacharya College of Pharmacy. It's my privilege and honor to introduce today's profound speaker, Dr. D. Josna Madam, working presently as teaching faculty in SKU College of Pharmaceutical Sciences, Sri Krishnadevara University, Anantapuram. So, Madam has completed his Bachelor in Pharmacy from Sri Padmavati School of Pharmacy in the year of 2006 and Masters in Pharmacy from Faculty of Pharmacy, Sri Venkateshwara University in the year of 2009. And PhD was awarded from Sri Padmavati Mahila Vishwavidyalaya, Tirupati in the year of 2021. And Madam has overall teaching experience, around 12 years of your teaching experience. And Madam, we are being actively involved in various pharmaceutical academic activities, such as a teaching, a training, evaluating, and the orienting of the students in different subjects which are covered under your pharmaceutics. And Madam has also published around 12 journals in well-reputed national and international journals. And Madam has been received a gold medal prize for the best outgoing student during the academic year 2002 to 2006. So with this brief information, so I would like to invite today's profound speaker, Dr. D. Josna Madam, for delivering the talk. Madam, welcome, Madam.
Kindly switch on your mic.
Am I audible now?
Yeah, madam.
Yes, Madam.
Yeah. Thank you, Praveen Kumar sir, for the nice introduction.
Yeah, welcome, Madam.
Sir, can I share the
madam.
screen?
You may share. You may share now. Madam. You are the co-host now. You can share.
So is it visible for all?
1 second, 1 second. Your screen sharing, 1 second. Yeah, madam. Yeah, madam. Excellent.
Just full screen, madam.
Make it slideshow, madam.
1 second.
If possible, press F5. Otherwise, make it slideshow. Yeah.
One second.
Oh, no problem. No problem. Yeah, madam. Excellent.
Is it clear?
Excellent, madam. Excellent.
Okay. Thank you, sir. So good morning to all the participants, and firstly to the principal madam, Dr. Swanlata madam, Dr. Dwarakananda Reddy sir. A very good morning to one and all the participants and the delegates who are over here for the session. And the topic of the session is design of experiments applied to pharmaceutical quality by design. And moving to the design of experiments, we all know knowledge is divine. Yes, and this knowledge can be a catalyst for change to researchers or academicians in today's highly competitive environment. And such a knowledge about a process or a product or a system, especially we researchers, it will be gained only through experimentation. And with that experimentation, there are so many problems for the researchers, and we have to get very decide research outputs that are delivered to the marketplace on time with minimum resources. So these are the key problems with more and more experimentations, and such a challenge has to be overcome by the researchers. But presently, researchers always will keep on updating day by day. With that, they need to study a large number of variables with minimum number of experimental runs. And such a thing will be happen with quality by design. And quality by design is old concept, and it was being introduced by so many eminent personalities. And one among them I picked up, that's the great Duran. He explained that the quality problems are related for a pharmaceutical product development generally is because of poor designing of the process. Although no matter how many tests we are performed, but if at all we are going for the poor designing of the product, that leads to very poor safety or efficacy. And there has been a recognition of this quality by design. It will be improved by merely increasing, and it is built into the product, and thereby it was developed. And the definition of this quality by design is it's a systematic approach to the pharmaceutical development, which begins with predefined objectives, and it emphasizes product and process understanding. Whereas process control, it should be based on very sound science and quality risk management. And there are few goals for this quality by design. We need to achieve meaningful product quality specifications, whatever we desire, or as per the protocol that we need to achieve. And it should increase the process capability and reduce product variability, and to increase the pharmaceutical development and manufacturing efficiencies. Very importantly, we have to enhance the cause-effect analysis and regulatory flexibility. There are few steps of pharmaceutical quality by design, and such steps could be done in 3 ways. Initially, by establishing quality target product profile, and in this, it is a summary of quality characteristics of pharmaceutical product that must be achieved to guarantee safety and efficacy. So there are so many considerations we should include, such as it should be intended use in clinical settings like route of administration, dosage form, delivery system, dosage strength, container closure system. Next step is identification of critical quality attributes. These critical quality attributes are physicochemical properties or microbiological properties of pharmaceutical product. It could be in process quality control test and finished product quality control test that must be within the specifications. So the examples we can select as critical quality attributes are, it could be assay, content uniformity or degradation products, drug release, dissolution, etc. In this critical quality attributes, we can able to include critical material attributes. Critical material attributes are physicochemical properties that input material must comply to the desired output. And critical process attributes, parameters. These critical process parameters are like mixing time, stirring speed, temperature. These also can be identified in order to achieve critical quality attribute, desired critical quality attribute. And step 3 is risk assessment. It is a systematic process of organizing the knowledge information to support the decision. So it could be done with the 3 essential elements, starting with risk identification, risk analysis, risk evaluation. Risk identification could be done from potential sources of hazards, and risk analysis. These will be associated with identified hazards. Risk evaluation could be done with qualitatively with the Ishikawa, that is fishbone diagram, and quantitatively with the failure mode and effect analysis. Right. So with this, we can able to assume that for getting a designed product with minimum number of experiments, we have to be very careful. In each and every step, we can able to see there is quality target profile, there is critical quality attributes, and risk assessment. So it's very difficult, or else we can go with the old traditional method. So we should be having a minute point of doubt, and that will be cleared off with this. So I'll take an example that an experimenter wants to, once again, an experimenter wants to determine the best settings of 2 factors, suppose A and B. Then we'll go with the old traditional method, that is 1 factor at a time. Some people will call it as one factor at a time. So this is the traditional method where we are following with the trial and error method. So in this, we'll take each factor is varied 1 at a time around its control level. So we can go with, we are assuming that we are taking 2 experiments where we are fixing the B factor and we are changing A factor at its control level A. And in the second experiment, we are fixing the factor A and we are changing the factor B at its control level again. So this would be the control levels 0, 0, 0. That will be A. This is the control level in, and for B, this is the control level. So if we are performing these 2 experiments, what happens? We can able to get a maximum variation is observed between the low and high levels of A, that is -1 to 0, and at high level 1 to 0. We can able to get a maximum variation in first experiment, and in the second experiment, there will be a minimum variation between both the levels for factor B. So for best results, the factor A is required to set its high level, but we are leaving as such constantly factor B. We are not changing anything in factor B. So if at all, the problem with the conclusion is that we can able to never determine the factors A and B, whether they are interacted or not. If they are interacted, it might be the response of A or at the same level. Which level of A or which level of B, we can't able to determine at all. For suppose if A and B interacted, the best results occur when the factors A and B are at the highest level. So this is if this conclusion we are giving, it is wrong. We can't exactly say at which level we can get the best results, either at the low level or high level. We can't say, simply we are saying at the highest level. So to overcome such kind of misleading conclusions, so the approach is came with DOE, that is design of experiments. It is a statistical tool deployed in the quality by design. So in this, it is a very simple approach where it rotates the points to the corners of the design space. So this here. So this is a factorial experiment. There will be so many different types of experiments in DOE. So where both the factors are varied together. So here we can able to determine individual effect of the factor A. So individual of the factor A, we can able to determine by comparing the responses of 2 points at two low level of A, that's -1, 1 and -1, 1, and also to the 2 points at high level of A and B. That's 1, -1 and 1, 1. So the effect of factor B will also can be determined with the same by comparing the responses of 2 points at low level of A, B, and to the 2 points at high level of A and B. Not only individual effects we can able to determine the effect of interactions of A and B by comparing with the diagonal points, So this we can able to determine with design of experiments. And this is the roadmap where I'm going to explain about the design of experiments applied to pharmaceutical QBD. That's benefits of design of experiments over one factor at a time. Experimentation. Basic terminologies, fundamentals of DOE, execution of experimental design. There are two case studies where I can explain this design of experiments, starting with benefits over one factor at a time. So if you are talking about the benefits, we have to already be seeing the disadvantages or pitfalls of one factor at a time. Hello. Okay, these are the pitfalls, limitations, where there is an estimation of factor effects is very less. And as I said in the previous example, I take a very simple example to explain about the disadvantages or pitfalls of one factor at a time, where if you are taking X is an event of stirring a cup of coffee and Y is an event of adding sugar to the cup of the coffee, the effect of factors X and Y will be the sweetness of the coffee. It depends on the levels of X and Y. So if individually we are performing stirring of a cup of coffee without adding the sugar, it won't give the sweetness for the coffee. So that interaction effect we can't able to find with this one factor at a time. So ultimately we can't able to get a good cup of a coffee with appropriate sweetness. If that causes some misleading optimal conditions for the process. So that's why the design of experiment has been recognized thoroughly. And the benefits is we can able to maximize the process knowledge with minimum use of resources, and we can able to provide accurate information in the most efficient way possible. We can able to identify factor interactions and characterize the relative significance of each factor. We can allow for the prediction of the process behavior within the design space. And very importantly, we can able to establish a solid cause and effect relationship between the critical process parameters and critical quality attributes. Thereby, we can enable to estimate critical quality attributes through appropriate selection of critical process parameter setting. Thereby, we can able to establish a proven acceptable range. And how the DOE is applied to the pharmaceutical QBD? DOE is the main component of statistical toolbox to deploy quality by design research, and it is a call for the adoption of rational, systematized, and cost-efficient strategies. So it is meant for products or process developed to evaluate all the potential factors simultaneously. So we already included the potential factors. It could be critical quality attributes. Both we can take critical material attributes or critical process parameters systematically. So we can get the results with the minimum time. And implementation invariably, it encompasses the use of statistical experimental designs where there will be generation of mathematical equations, and with a complete picture we can able to get the variations of the response as a function of factors. And according to ICH Q8 guidelines, so it creates a favorable environment for the use of DOE because of dramatic increase nowadays of relevant scientific works and industrial applications. If you go on checking for any updated information for doing a research, there are so many papers on DOE, not only in the pharmaceutical in pharmaceutical analysis, and in healthcare also, and in engineering also. So there are so many disciplines where they are taking the advantage of this software. And it is very user-friendly software also for facilitating the construction and for analyzing the designs. So that's why the DOE is the key to the magic kingdom of quality. We'll quickly move on to the fundamentals of design of experiments. So there are three basic fundamental principles we have to know before going for the execution of the design of experiments. First one is replication. Replication is simply once we are getting the once we are giving the input to the DOE software, it will create one design matrix, and it includes so many experiments which are repeatedly coming with the same factors and same levels. That is only to estimate the experimental errors, how much the errors will be happening by the experiments, so that we can able to assure very accurate and precise outputs or estimates. Next one is randomization. In this randomization, it is a process of running experiments in a random order to avoid subjective decisions or bias, so that we can able to minimize the effects of uncontrollable changes. Blocking. In this blocking, the third fundamental principle is blocking. In this process, we can group the trials and where the experiments into subgroups or blocks. So with this basic fundamentals of design of experiments, we can go with execution of experimental design. So this is the key concept of experimental design. So in this picture, you can see that the controlled and uncontrolled factors, these both factors are determined to reach the target, that is the quality. And these controlled factors are called as inputs or factors or independent variables, which could be represented generally with the letter X. So how many number of factors we are selecting, based on that we will give X1, X2 and X3 were here. So that independent variables already in steps of pharmaceutical quality by design, I already explained identification of the critical quality attributes. So we should be very careful in identifying that. We can go for some preliminary trials, or we can have a very sound knowledge by means of historical data. By that, we can able to select the controlled factors. And find these are outputs, the quality where we are reaching the quality, that is the output. We can name it as responses, and these are dependent variables, and these are dependent on the inputs. So these could be the yield or dissolution, impurities, etc. And what are these uncontrolled factors? These are nuisance parameters, which will be, some will be measurable and some will be non-measurable, like variability in equipment performance and environment operator. So this all relationship between the inputs and outputs is cleared with the help of a mathematical model that is given in the form of y is equal to f of x. So by this way, I can in very briefly we can say this is what design of experimentation will do. But there are few steps to get all these things. And those steps include is step 1, we have to set a proper objective. And in step 2, we have to go for selecting factors with their levels and responses. Step 3, we have to select a suitable experimental design. Step 4, we have to conduct the experiments accordingly as per the generation of the design matrix by the software, and we have to collect the data. And based upon the results generated by the software, we have to go for selecting or developing a suitable mathematical model. Once it is selected, we have to go for analyzing the results. And finally, which model we are selected, whether it is valid or not for the whole goal that we have to validate. So these are the 7 steps for executing the experimental design. So we'll quickly move on to the step 1. In step 1, I'm taking the basic example of study of formulation variables. This is an example only to explain the step 1 and step 2. So we are taking an example where we are going to study the effect of formulation variables influencing polymeric microparticles by experimental design. So the first step, setting the objective, you should hit right on the target. So a very strong attempt is to be made to explicitly ascertain the drug delivery objective. For example, in this example, the effect of formulation variables on diclofenac sodium loaded microparticles is being determined in order to obtain a sustained release system. So the goal should be very right. And for that, we have to go with certain objectives. And in the second step, so here we are going for preparing the microparticles containing diclofenac sodium using oil in water single emulsion solvent evaporation method. And in this method, we are taking 250 mg of diclofenac sodium. And this diclofenac sodium was mixed with dichloromethane, 10 ml and methanol. Then we are adding the proportions of methyl cellulose and polymer, and it was dispersed in 100 ml of distilled water, which is containing varying amount of PVA, which is an emulsifier. And this has been prepared. The microspheres has been prepared by solvent evaporation technique. And from this method, selection has been done with a proper historical background or a good sound science of literature review. They select that amount of ethyl cellulose and amount of polyvinyl alcohol will be having a strong effect on encapsulation efficiency. And then they have to select the levels for the factors. They have to select levels for the factor. And they selected 2 levels, low level and high level. So these are the levels. So this is how they, in step 2, they need to select factors and their levels and responses. In step 3, selection of suitable experimental designs. To map the responses on the basis of study objective, responses being explored and number and type of factors and factor levels, there are few designs available: factorial designs, mixture designs. In factorial designs, one can allow to study a wide number of input factors with reduced number of experiments. So with a less number of experiments, we can able to study more number of factors. In mixture design, this is highly recommended in drug delivery systems with multiple excipients. Factorial designs, in factorial designs, again, there will be screening designs and optimization designs. In screening designs, there are 3: 2-level full factorial, fractionate factorial, Plunkett-Berman, or we can name it as Hadamard designs. So in this table will explain how many number of experiments each design will perform with how many levels and how many factors and what are the limitations. Two-level full factorial design, the number of experiments will be 2 to the rise of k, where here 2 is the levels and k is the factors. So there will be 2 levels and factors could be from 2 to 5. But the problem with two-level full factorial design is it relies on very large number of experiments. So we have to, 5 factors means we have to depend on more number of experiments. So we are moving to the next fractionate factorial designs. Here the number of experiments is 2 to the rise of k minus p. And as we know, k is factors and 2 is levels and p is the number of the generators. And RES is the resolution, I'll explain now. And levels is 2 and factors is greater than 4. What happens in this fractionate factorial designs? The main effects and the interaction effects will be aliased or confounded. That means if 2 factors or interaction terms are at the identical levels throughout the experimental designs. So this happens in fractional factorial designs. For example, if you are going with 2 to the rise of 3 minus 1 resolution 3, the factor x3 will be interacted with the second order interactions x1 and x2. Whereas with 2 to the rise of 4 minus 1 4 resolution, the main effect x4 will be interacting with the third order interactions of x1, x2 and x3. Whereas in fifth resolution of fractionate full factorial design, the main effects will be aliased with the fourth order interactions and second order interactions are aliased with third order interactions. So these are the limitations where the usage of this fractionate factorial designs is very minimum. Moving to the Plunkett-Berman, the number of experiments will be 2 to the rise of k, where the levels will be 2 and factors will be greater than n minus 1. So it allows one to study factors, n minus 1 input factors, where n is the multiple of 4 with n experiments. So these are the screening designs, and this is the pictorial representation of the fractionate factorial design. That's example. The design is for 2 to the rise of 3 minus 1 fractionate factorial design. Here the design will be as peers. Coming to the optimization designs. So these are very much familiar. There are 3-level factorial designs where the experiments will be 3 to the rise of k. And the levels will be 3. Factors will be either 2 to 3. Central composite design, and the advantages of 3-level factorial design is it is always used when 2 or 3 input factors are need to be studied, because it will generate an increase in number of experiments. And in central composite design, the number of experiments will be 2 to the rise of k plus 2k plus c, where the levels will be 5 and the factors will be the 5 factors. Mostly the central composite design will be used for nonlinear responses. That means it always requires second order models, and it is more advantageous, and it will synergistically combine the advantages of factorial designs and star designs. And Box-Behenken design, where the number of experiments will be 2k, which will be multiplied with k minus 1 plus c, and the levels will be 3, and the number of factors will be 3 to 5. So these are special type of 3-level fractionate factorial design, and it will overcome the first order and second order response surfaces, which is very cost effective than 3-level full factorial designs. These are the factorial designs, and these are the pictorial representations for 3-level factorial designs, central composite design, and Box-Behenken design. Mixture designs. So generally in factorial design, we are initially we are selecting the factors and their levels, but in mixture design, it is aimed to rationalize the use of each constituent and its proportion. So here we are adjusting the proportion of the excipients. In this mixture designs, the component proportions are not independent of each other. The proportion of one component increases and the proportion of one or more of the remaining components should decrease. And normally, the relation between the responses on the component proportions will be in this way. There will be 2-component mixture design and 3-component mixture design, where in 2-component, only 1 factor level can be independently varied. In 3-component, 2 factor levels can be independently varied. And this will be the linear model, and this will be the quadratic model or nonlinear model. It is very much useful for suitable for formulations in which constraint imposed on some combination of factor levels. And in step 4, we have to go for conducting experiment. As per the design experiment software, what it is executed, we have to perform in such a way. We should be keeping as the factors constants. As per its design matrix, we have to perform the experiment. And experimentally, it should be according to the chosen experimental design, and we have to evaluate the selected response variables, and we have to incorporate into the experimental design software. Once we are going to incorporate, we have to go for developing a mathematical model, which is the basic scope of design experiments. It is a important way where we can able to describe the factor and response relationship. So model development, it won't happen in a single step. So we have to go for performing so many number of things we have to measure and we have to interpret, we have to include. Initially, we have the design expert software will apply ANOVA, and from that we have to go for selecting the mathematical model, which is used to compare the variability due to treatment with the variability due to residual error. And once we are selecting the mathematical model, whether it is a first order or second order or quadratic model, whatever model we are selecting, then we have to go for including or excluding the coefficients of the quantitative factor terms, that is individual factors x1, x2, x3, or interaction terms x1, x2, x1, x3, and x2, x3, and quadratic terms x1 to the rise of 2, x2 to the rise of 2, and x3 to the rise of 2. The inclusion or exclusion is based on the probability values for each coefficient regression term. So the coefficient regression term, if we want to include, it should be lower than 0.05. If it is higher than that, we can say these terms are insignificant and we will exclude it from the mathematical model. So generally the models used to describe the responses will be either first order, second order, or very rarely it will be third order polynomials. And for this, the models will be developed with the regression analysis. So they will estimate the coefficients for quantitative factors if it is first order. If it is non-linear, it will go with multiple regression analysis, where there will be estimate of more factors, interactions, and higher order terms. So we are preferring multiple regression analysis. And in this multiple regression analysis, the model will be adjusted based on determination coefficients. So these are the determination coefficients, where R2 adjusted R square, predictive R square. R square is the proportion of variance. It is the proportion of variance in the output response that is predicted from the input factors. And this R square always increases by adding new terms. If there is in the model, if that we are including any new terms, the R square value will be rising high. And adjusted R square is modified version. It should be adjusted based on the number of terms in the regression model. It increases if at all any new term improves the regression model. If not, it decreases. And predict QR square, it indicates how well the regression model predicts the output responses for new observations. And both this adjusted R square and predictive R square are always lower than the R square. And not only these determination coefficients, we have to check the lack of fit also. Whether there is a variability, we have to compare it with the variability due to random pure error, so that we can able to assess the regression model adjustment. And there should be one key line here. We have to note that the error provided by the regression model, lack of it, is significantly lower than the random pure error. Then only, that is the probability value should be greater than 0.05. Then only we can say the regression model is well fitted. And this lack of fit error is estimated only if there are replicas. Usually the replicas will be, if the design is having center points, then only such kind of replicas will be there in the design matrix, which is generated by the design experiment software. And analyzing the results. So it could be done by various graphs, which are very, very useful for analyzing the results. There are Pareto charts, which it will allow to select the very important input factors and their interactions. And there will be graphs available for the main effects to determine main effects and interaction plots. These provide the information on how the responses will have an effect with varying levels of factors. And half probability plot or normal probability plot. So these will be plotting the data by taking the theoretical normal distribution in such a way that the point should form an approximate straight line. So if at all the points will be deviating from the straight line, it indicates if these are departing from the normality. Whereas in response versus prediction, so these we can able to identify any interaction or involvement between the factors. So these graphs will help in identifying individually, each graph will be having its own application, its own purpose. And finally, validation of the model. This validation can be done by different methods. One is graphical method and mathematical optimization, artificial neural networks, extrapolation outside the domain. In graphical optimization, there will be brute force search or overlay plots or combined contour plots or canonical analysis. So in brute force search, all the possible combinations of the independent factors for their outputs will be analyzed, will be checked, and they will give a set of acceptable limits for both, and they will be having an exhaust search to identify a final feasible formulation, feasible search, so that they'll generate an optimized formulation by checking all the possible independent factors and their effects on the response variable. In overlay plots, we have to give a minimum and maximum boundaries for our desired responses, so that by superimposing this by dimensional responses, there will be certain overlay plots will be generated by the design experiment software, so that from that we can have to choose the design space and whether our desired output is within the levels of the design space. And canonical analysis. So the predictability of each extracted components of the criterion set of variables, it will be indicating. And mathematical optimization, generally, it could be done by associating each response with its own partial desirability function. So we'll go with the 1 point by giving highest value of desirability, which is treated as optimum. And then from the contour plots of desirability surface around the optimum, it should be studied. And it should be combined with the contour plot of most important responses. So these are graphical mathematical optimization and artificial neural networks. This will be machine-based computational technique, so which will simulate one of the neurological processing abilities of the human brain, and extrapolation outside brain This is the steepest ascent method where the optimum is searched outside the experimental domain by extrapolation. Case study. So what I selected the example for explaining the step 1 and step 2, the same example I am here included in case 1, that is study of formulation variables influencing polymeric microparticles by experimental design. So we already had an overlook of this where we selected the factors as amount of ethyl cellulose and amount of as A and amount of polyvinyl alcohol as B, and the output, that is response, as encapsulation efficiency. So these are the 2 steps already we had an idea, and in step 3, the selection of suitable experimental design. So here 2 levels, low level and high level, which is indicated by -1 and +1. So 2 factors and 2 level experimental design matrix. So this will be given in coded levels where there will be 2 to the rise of 2, there will be 4 experiments will be performed, and this is generated by the software. And step 4, we have to conduct the experiments and we have to collect the data and we have to incorporate into the design experimental software. That is encapsulation efficiency. We performed for this 4 trials and we got these values and we incorporated into the experimental design. And what happens in step 4, we have to go with a mathematical, we have to go for selecting the suitable mathematical model with the help of ANOVA. So this is the ANOVA table where it is created. So in this mathematical model, it clearly gives the relation between the factors, that is amount of ethyl cellulose and amount of polyvinyl alcohol on encapsulation efficiency, where here the positive sign of factor A indicates that, so if there is increase in the amount of ethyl cellulose, there will be increase in the encapsulation efficiency also. And the negative sign of factor B indicates that is the amount of polyvinyl alcohol. It indicates that if there is increase in the amount of polyvinyl alcohol, there will be decrease in the encapsulation efficiency. Fine. And here we can see these 2 coefficient terms. We are included in the mathematical model because here these are having the probability value less than 0.05. That's why we are included here. And the next thing, not only we have to perform, we have to check the ANOVA table and we have to go with this lack of fit values. We have to go with this R-square values also, determination coefficient values. So the R-square value will be 0.993 and adjusted R-square 0.9978 and predicted R-square 0.9884. So it's clearly indicating that the adjusted R-square and predicted R-square will be lower than this R-square. So we can say the lack of fit is significant. And next 6th step is we have to analyze the results. We have to analyze these results to graphs. So Pareto charts, I already said these are standardized Pareto charts where we can able to estimate the effect of the parameters on responses. So one can allow us to check the statistical significance of this experimental designs. So whereas it consisting of bars with lengths which is proportional to the absolute value of the estimated effects. So that will be divided by the standard error, and this all calculation will be done by the student's teachers in the software. And from this graph itself, it clearly indicates the amount of ethyl cellulose and the amount of efficiency of the polymeric microparticles. and half normal probability plot. In this half normal probability plot, we can see the departure of the points from the straight line. So it just indicates that these are deviating or departing from the normality. And coming to the normal residuals, normal plot of residuals, where we can estimate all the points will be indulging in the straight line, showing that the linearity and interactions and involvement between the factors is there. And contour plot and 2D contour plot and 3D response surface plot. So from these two plots, we can clearly identify that with the increase in the ethyl cellulose, so the high level of factor A will be having the high level of factor A, that is amount of ethyl cellulose, will be increasing the encapsulation efficiency. At the same time, with the high level of polyvinyl alcohol, there will be decreasing encapsulation efficiency. So this is about case 1 and case 2 study. In case 2 study, I picked up this example, that is formulation and optimization of sustained release dalfampridine node and microspheres using natural polymers. And in this step 1, we have to be right to the target. So we are going to develop and optimize sustained release dalfampridine node and microspheres using natural polymers as release retardant materials. And this is where we are preparing the microspheres by preparing a blend solution of the polymers. We are making into a homogeneous dispersion using mechanical stirrer, and then we are adding the drug to that dispersion, and we are making it again homogeneous dispersion. And once it is prepared, this polymeric solution was added dropwise to the calcium chloride solution by using hypodermic syringe. And we are retaining the microspheres for 30 minutes for curing and to produce spherical region microspheres. And then these hardened microspheres are filtered and it will be rinsed with distilled water thoroughly and dried. And it will be stored for the further analysis. In step 2, there is a wide literature review was been collected for selection of independent variables and dependent variables, that is factors and responses. And here A, B, and C, that is polymer ratio and agitation speed and calcium chloride. Here the factors are represented with the terms A, B, and C, and dependent variable, drug entrapment efficiency. And in step 3, selection of the suitable design, experimental design. Here we selected central composite design, where we are taking 2 level 3 factor central composite design, and this is in combination with the response surface methodology for optimization of sustained release dalfampridine node and microspheres using natural polymers. And in step 4, as usually, we have to go for conducting the runs as it is generated by the design experiment software. So here we are getting around 20 experiments, so which are framed with 8 factorial points, 6 axial points, and 6 center points. And once we are performing all the experimental runs, we incorporated the results that are fitted into the design experiment software. And in step 5, that is selection of model development. In this model development, we gone for the first step and ANOVA, where in this ANOVA, we are selected the suitable mathematical model, and these mathematical model, this is the generated mathematical model by the software, and we pick this quadratic model. This is quadratic model, where it includes the interaction terms and quadratic terms, but here this model is reduced in this way. Why it is reduced to this model with less number of coefficient terms mean, see here, we are getting not all the terms significant here. So there are so many terms which are crossing the 0.05 probability value. So if it is crossing the probability value, those are finded as non-significant. So once they are finded as non-significant, we have to remove the insignificant terms on the basis of the statistical analysis. And then we have to generate a reduced quadratic model. And this is the reduced quadratic model, that is R1 is equal to 86.83 plus 2.048 plus 9.91C and minus 2.63A raised to 2 minus 1.7 B raised to 2. And in this reduced quadratic model, we have the significant terms with the signs here. So with this signs, we can be able to identify that factor A and factor C will be having the synergistic effect for entrapment efficiency. Whereas the interaction quadratic terms A square and B square both are having the antagonistic effect, where the increase in A square and increase in B square will be decreasing the entrapment efficiency. And coming to the step 6, so analyzing the results with the help of the normal plot of residues and the graphs with predicted and actual responses, and we here normal plot of residuals. There are maximum number of points are within the straight line, that says it indicates it is all the points are within the norm are in the normal. They are not departing from the normality. And coming to the predicted versus actual, so it shows the rare points where the few levels of different factors are deviating from the straight line. And here, as I already said, that from the equation and with the signs, we can clearly identify the individual impact of the factors. And not only that, we will be having a significant positive effect of individual factors and significant antagonist effect of the squared impact of the factors. So hence, we can say that drug entrapment efficiency will increase with increase in ANP and will decrease with synergistic rise in quadratic terms A square and B square respectively. Thus, we can say the optimum levels of A, B, and C are very important for obtaining the desired values of drug entrapment efficiency of dalfampridine load in microspheres. And this is the perturbation plots where we can get from the response surface methodology, where from these plots also we can clearly determine and we can visually represent or we can understand the factors A, that is the polymer ratio, and factor C. Both will be having a significant effect in altering the response of drug entrapment efficiency. So with the increased levels of A and C, it results in increase in drug entrapment efficiency. And whereas the effect of the squared terms of A and B on drug entrapment efficiency was observed to be slightly curvilinear, showing that non-linear effect. So with increasing levels of quadratic terms A square and B square, it results in decreased drug entrapment efficiency. And these are the 3D and 2D contour plots. And these shows the curvature effects of the polymeric ratio of A, that is polymeric ratio, and B is the agitation speed. As the polymeric ratio increases, so this polymeric ratio increases, there will be increase in the drug encapsulation efficiency. And simultaneously, there will be decrease also followed by a decrease. So hence we can see a parabolic effect of both factor A, that is polymer ratio, and B, that is agitation speed, on drug entrapment efficiency we can observe. So maximum entrapment efficiency was observed at the intermediate levels. So maximum entrapment will be observed at the intermediate levels of polymer ratio and agitation speed. So the similar results will be observed in this 2D contour plots. And in step 7, this is the numerical optimization technique has been done by surface response methodology, where we have to give the maximum and minimum boundaries for each factors. For each factor, we have to give as the input to the design experiment software. After that, it will generate one optimized formulation with a set of factors. So with this set of factors, that is polymer ratio 3.33, with this ratio, we have to go for performing an optimized formula. And this B, that is the stirring speed, we have to choose as 763 RPM. So as this is in point decimals, we can go with 760 RPM. And concentration of calcium chloride 0.955. So if we are performing this optimized formulation, we have to get the response with this limit. So this boundaries maximum minimum we have to incorporate into the design software, and once it has to be within this, then drug entrapment efficiency should be 85.2967. So this is the overlay plots or desirability plot, which is generated by the software. And we have to perform this optimized formulation with this optimum condition generated by the software. And if once we are performing this with this optimum conditions, we are getting so this is 85.29. It is generated by the software. And we got this. So this 86.09 is the value is achieved with this optimum formulation for the drug entrapment efficiency. So we have to calculate the relative error. So as the relative error is very low, so the low magnitude of relative error will confers the predictability and robustness of the software in optimization of dalfampridine loaded microspheres. So this is how we have to go with step-by-step analysis for any design we are selecting, if it is two-level factorial design or if it is central composite design. So if we are following in a step-by-step manner, we can able to understand easily without any confusion, and we can get the most desired responses with the factor levels which are generated by the software. So this is the paper what I published for my PhD work, that is novel tamarind seed gum alginate based multiparticulates for sustained release of dalfampridine using response surface methodology. And coming to the conclusion, so with this rising awareness of statistical thinking, the use of experimental design has been rapidly permeating in so many disciplines of medicine, engineering, technology, industry, and research, and in health sciences. So the DOE and quality by design were widely applied today in so many diverse technologies. Hence, DOE applied, design of experiments applied in pharmaceutical quality by design can strongly facilitated by the implementation of techniques. What I described like screening designs, it could be optimization designs or mixture designs for optimizing not only the drug formulations, but also the process which leads to the development. So that's, I'm concluding. And these are the references. And I want to thank the organizers and Principal Madam, Dr. Swanlata Madam, Dr. Dwarakananda Reddy sir, for giving this opportunity to share about DOE on this online platform. And I want to thank my principal and director for encouraging me in each and every event. And I also want to thank the teachers right from my UG to PG, where I studied, like Sri Padmavati School of Pharmacy and Sri Venkateshwara University and Sri Padmavati Mahila Vishwavidyalayam and my guide. So thank you all my teachers. I would like to thank all my teachers for their teachings and efforts. And you have put into me a better person in life. And now I'm today because of all of you. And thank you one and all.
Yeah, thank you, madam. Thank you for your enlightened presentation, madam. And your presentation was very clear and also been very informative, madam. And as a token of appreciation, so we have been presenting you a virtual e-certificate, madam. So kindly accept it. And the same will be mailed you after the session, madam.
Thank you, sir. Thank you.
Thank you for your presentation and accepting for delivering a talk in phase 3 of ASCT sponsored STTP, madam.
I also want to thank all the participants and the delegates who are very patient in listening and hearing the topic throughout the session. And once again, I want to say my deepest thanks to the organizers and the principal sir, Principal Madam Swarlata ma'am and Dwarakananda Reddy sir, and all the faculty of the Annamacharya College of Pharmacy. You did a wonderful job. Thank you.
Yeah, thanks for your appreciation of whole the team of Annamacharya, madam.
Thank you, sir.
Thank you, madam. Dear participants, so the next session will be commence exactly 2 o'clock in the afternoon. So kindly log in without any delay exactly by 2 p.m. in the afternoon session, which is also as one of your informative session to be delivered in the afternoon session. Over to the host, Pradeep sir.
Thank you, sir. Thank you so much. The session will be start exactly by 2 p.m. Catch you by 2 p.m.
Sir, good afternoon, sir.
Good afternoon, sir.
Sir, good afternoon, sir.
Good afternoon, sir. Am I audible?
Yes, sir. Clearly, sir. Clearly audible.
Oh, great. Okay.
Audible, sir.
Hi, Derek.
Good afternoon.
Great, great.
Yes, thank you. Hearty welcome to our ASCT sponsored short-term training program, sir, for faculty members.
Okay.
Pradeep?
Sir, sir.
Deepa?
Sir, Deepa madam there, sir. Deepa madam.
Start, Jay Pradeep.
Good afternoon. Good afternoon to respected Principal Madam, Dr. P. Sonalata Madam, and Dr. P. Dwarakananda Reddy sir, Program Coordinator, Short-term Training Program, Phase 3.
Pradeep, start, Jay.
Sir, already started, sir.
Start, Jay.
Good afternoon to respected faculty members. Today, speaker, Venkatesh sir, respected Principal Madam, Swanlata Madam, and the Vice Principal and the coordinator of this program, Dr. P. Dwarakananda Reddy sir, and all the participants of today's short-term training program, Phase 3 afternoon, basics. So, I would like to invite today's speaker, Venkatesh Ramdas sir. So, on behalf of Annamacharya College of Pharmacy, I, Mr. Y. Pradeep Kumar, Assistant Professor, Department of Pharmaceutical Chemistry, and it's my immense pleasure to invite Venkatesh Ramdas sir. Sir completed Doctor of Philosophy in Pharmacy, Jadavur University in the year of 2012. Sir completed Master of Pharmacy from Birla Institute of Technology and Science, BITS Pilani, Rajasthan in the year of 1998. And Sir completed his Bachelor of Pharmacy from Madurai Medical College, Madurai, Tamil Nadu in the year of 1993. Sir presently working as the General Manager, QA Department at Hipro Healthcare Private Limited, Chennai, India. Sir previously worked as General Manager in the Sai Mira Innofarm Private Limited, Chennai, India, and also Captain Point Laboratories Limited, Chennai, India, and he's worked as a Vice President Formulation. Sir cleared international audits like PIC, that is Pharmaceutical Inspection Corporation Scheme, Colombia, FDA, Guatemala, Tanzania, Nigeria, Kenya, Jajibonia, and CDSCO, India. Sir mainly focused on research and Sir having 7 international publications, and Sir co-authored a book on natural chemistry. So with this brief note, I would like to invite Ramdas sir to enlighten the today's session. Thank you, sir. Thank you so much, sir.
Thank you, sir. Good afternoon, all. Today's topic, you had a very nice session in the morning, 2 sessions you have. And in the afternoon, we are going to discuss about the quality by design. This topic, I just want to tell you, like a statutory warning we normally give in the cigarette, you just mention, cigarette smoking is injurious to health. So this topic, what we're going to discuss today, is going to be a very dry topic. It's very dry. And unfortunately, after the lunch, you are having the discussion on this topic. So naturally, the combination of both the factors is likely to lead you into a sleep. So what I request you, participant, is to keep your eyes, ears, and senses very active to get the discussion of this, to go into the discussion of this thing. Before delving into this topic, I just want to tell, we will be refreshing all the basic B.Pharm aspects what you studied, because whatever you study in the B.Pharm is going to be the construction of this thing is going to be M.Pharm, and PhD you're not going to study anything. The fundamental things are going to be constructive phase. So with this background, we go into the topic, quality by design. I just want to ask you, what is quality? Can you flip the next slide, please? What's quality? Next slide, please. Yeah. I'm in the online session. Let me give the question. I'll ask the question myself, and I need to give the answer myself. Quality is fitness of purpose, right? So how do you estimate quality? Quality can be in the form of food. What you're eating is a quality food. You can eat a quality, you can sense a quality, you can smell a quality if it's going to be a beautiful perfume, or you can feel a quality like you wear a good cloth, the textile, what you feel, you can feel the quality. Maybe the footwear you're purchasing, that can give a good feel. So the quality is embedded and pervaded in all the aspects of our life. So how do we define the quality? Quality is just a fitness of purpose. So, but what is, how do you estimate a quality? How do you go with the quality? Like I give normally an analogy. You want to cut a tree. There are 3 axes given to you. One is going to be an iron axe. Second is going to be a golden axe. Third one is going to be an aluminum axe. Which axe will you prefer for cutting a tree? Again, unfortunately, I have to pose the question and I need to answer the question. So you choose an iron axe or golden axe or an aluminum axe. The choice is left to you. So normally we choose an iron axe. Okay. Why iron axe? Because that is fitness for the purpose. That's where the quality stands. Golden axe doesn't serve the purpose. It can cut it. You can cut the tree, but it's going to be very costly. Aluminum axe is very malleable and ductile, so it bends when you're cutting a tree. So the right choice is going to be your iron axe. So what is quality? Quality is the fitness of purpose. So one part of the title we have discussed. What is design? Design is the construction. You're going to construct something. You put a concept into it. You design it. you construct it, building the things that forms your design. So we are discussing this in the context of a formulation. You can use the quality by design for analysis also, and you can use it for formulation also. So this will be, this concept is used only in the developmental stage of a formulation or an analytical development. Well, so I just rattle the definition because I don't want to go deep into the slide, because going into the slide at this time, 2 o'clock in the afternoon, is going to be even more, sleep inducing. So we go more into the discussion. The first concept is going to be the risk-based formulation development, QBD, to design a quality product and its manufacturing process to consistently deliver the intended performance of the product. So the objective of quality by design is to construct a product to consistently deliver the intended performance of the product. So this is the intention of quality by design. It is intended to increase the product and process understanding and thereby decrease the patient risk. So when you are constructing the product, you understand the process that is associated with making the product. So that way you are reducing the risk associated with the product development. Okay. So the other one is quality by design is a regulatory requirement. So what is the regulatory requirement that is telling? We have an ICH. Okay, ICH is a regulatory body, a global regulatory body. It is stating in ICH Q8, it's asking about the QBD. It is talking about the QBD. European Union has already embraced on QBD, and also the US has gone on the QBD. Many regulators are asking for the QBD. Next one is a risk-based management. There also the QBD is also involved with the risk analysis in the development of the formulation. So the risk-based development is mentioned in the ICH Q9. The risk analysis is more accepted in many of the regulatory bodies, especially Europe. But US is going to impose that risk analysis in future ongoing years. They'll be going for the risk management. Then process validation, it is mentioned in ICH Q7. They are also asking for the process validation. The phase 1 of the process validation involves the QBD. It's associated with the QBD. So in both all the three concepts, the regulatory wise, they are requiring the the QBD. Well, in the case of QBD, you need to understand three concepts. One is going to be QTPP, CPP, CQA. What is QTTP? That is Quality Target Product Profile. Then CQA, Critical Quality Attribute or Critical to Quality Attributes. Then CPP is Critical Process Parameters. So these three are going to be the very important, three important concepts. If you understand these three concepts, you are almost, you're thorough with the QBD concept. So I'll try to lucidly, I try to take world analogies and try to put this three concepts into it. So kindly open your heart and mind just to listen this concept. After familiarizing this concept, next things will be very easily, lucidly, it will be going. So what is QTTP? Quality Target Product Profile. So now I'm looking for a girl for my marriage. This is an analogy. Kindly take it as an analogy point. So what I'm asking is, I want a first choice I'm giving to the person is like, I need a beautiful girl. The girl must be very beautiful. Second thing, what I ask is, the girl must be filthy rich. Okay. Third condition for me is, she must be knowledgeable. The girl must be very knowledgeable. Fourth one, this girl must be very kind. She is very, very kind. And in the world, in any part of this world, can you find a girl with all the four characters? It's very next to impossible, right? Well, okay. So this is the character I'm fixing for finding my life partner. Now I'm taking for developing a drug, a generic drug I am going to develop. Already the drug is available in the market. and I'm going to copycat the drug and I am going to market, make an ANDA formulation. What is it? ANDA? Abbreviated New Drug Development. I'm going to take up. For this aspect, I need to identify the ideal characteristic I need to put in my developmental drug. The drug, what I'm going to develop, what is the ideal characteristic of the drug? So I need to have someone to compare. Maybe if I am a girl, I just compare with, sorry, if I am a male, I compare with the Shah Rukh Khan or Salman Khan or Amir Khan or something like that. I try to imbibe the style of this thing. I just try to put the same shirt, coat, all this thing, hairstyle, everything. I just keep. So I try to mimic them, imitate them. In the case of development, generic development, I need to take the concept and I need to go with the attributes that is present in a already formulated drug, which we call it as a RLD. RLD means reference listed drug. Okay, from the reference listed drug, I need to identify the ideal characteristic. What sort of a color is there? What sort of a shape is there? What characteristic I need to borrow from those things, and I need to put it into my drug which I am going to develop. That is called the QTPP. So now I go to the definition. The quality target product profile is a prospective summary of the quality characteristic of a drug product that ideally will be achieved to ensure the desired quality, taking into account safety and efficacy of the drug product. You don't understand, right? I too don't understand. But you take the analogy into it. What is QTPP? I am going to develop a drug. I take the ideal characteristic of the drug that I require to make. Where do I take from? I take it from an RLD. Why do you do this thing? To account for the safety and efficacy of the drug that I'm going to make. So this QTPP is an essential element of a QBD approach and forms the basis of design and generic product development. We go for the next slide. So we went with the QTPP. Next one is going to the CPP and CQA. Okay. So earlier slide, sir. Earlier one. So initially identify the quality target product profile from the RLD. Well, I understood that concept. I'm going with this thing. Can you please go to the earlier slide? This can be done by characterizing the RLD. Intended patient's drug will be given label of the RLD, and many of the aspects I'll be taking for constructing my QTPP. Prepare the critical quality attributes. What is this called? Critical quality attributes based on the severity of the product will cause if it fails to meet the quality attribute, meets the quality attribute. Okay. So we have another two terms. One we have completed is QTPP. Second is going to be a CQA. CQA means critical to quality attributes or critical quality attribute. So what is this critical quality attribute? Now you have found the girl. Okay, the girl is there, and they are arranging you to. They are asking you to. Am I audible? Hello. Hello.
Audible, sir.
Yeah, sorry.
Audible,
So
sir.
you want to check whether the characteristic that you have identified earlier asked the girl is having or not. So you asked for she must be filthy rich, she has to be very educated, she must be beautiful, and she has to be very patient to whatever idiosyncrasies you have. So now you want to text the girl. Now the girl is sitting in front of you. You can assess the beauty of the girl by the look. It is a skin deep. You can assess from that one. Next one, what you want to assess? If she is filthy rich, that you can assess from what she is wearing, or your about to be father-in-law is going to present as a dowry. From that you can have this thing. Okay. Then the degree you can assess by speaking about her, like she's studied in economics or something, or stockbroking or something. You can ask her what is short straddle, what is long straddle, what is a bull run, what is a bear run, what is a bull belt. All these things you can. She will be very eloquently. She will be explaining you. So you can assess this girl is really academic oriented or practical oriented. All those aspects you can conclude from them. And last one you want to assess her whether she is patient. So you want to pinch that lady, a girl in front of you. Will she be okay? Will she be accepting the thing? She's going to slap you. If she doesn't slap you, she's going to suit for the fourth condition also. Unlikely expect you will get a return from the girl if you're going to pinch her or slap her mildly. So this is the way you're going to assess. The same way you're going to do it in the case of a drug. You designed a drug, you identify a drug. How do you identify the characteristic of the drug? Like you identify the girl's character, you want to do this thing, you identify the QTPP of the product, what you want to match. Second one, if the QTPP is matching and you are designing the drug, it has to be characterized. How it will be characterized? You will be characterizing those attributes through a critical to quality attributes. You will be measuring the critical to quality attributes. In the case of a drug, what are the CQAs you will be doing? What are the critical attributes you will be measuring? Most of you will be knowing the analysis. They'll be taking assay, dissolution, RS, related substance, or any other thing like dissolution, maybe the loss on drying, moisture content, many of the parameters that is coming, you will be taking into account for measuring the drug's attribute. That we call it as a critical quality attribute. We'll be seeing in more in detail this case. So, my dear friends, we have gone through two characteristics. What are the two attributes, two things we have seen? QTPP. So, fairly you must be knowing about what is QTPP. Second is critical to quality attribute. What is critical to quality attribute? There's nothing but the way you're going to measure the drug, where you're going to identify the efficacy of the drug in a laboratory setup. Okay, so that forms the CQA, critical to quality attribute. Two definitions or two things, concepts you have known. The third concept is going to be your critical process parameter. What is critical process parameter? My dear friends, I'm not a good cook. However, we'll take up an analogy of making puri. You're going to make a puri in your house. Okay, you are a bachelor, you're going to make a puri. What do you take first? Yeah, you'll be taking atta. What atta I'll take an atta from Pillsbury or Annapurna atta, whatever atta that is there in the market, whichever that is going to suit my fiber content that is there present in the atta, that is going to be a good puri from my old history. I'm going to experience. I'm going to take the particular atta. What I'll be putting? I'll open the pack and put it in a big vessel. Okay, open vessel. I'll just put it. Next thing, what I'm going to do? I'll be putting some salt into it. Yes or no? I'll be adding some salt into it, sprinkling some salt. After that, I'm going to gently with my fingers, I'm going to knead this dry in this atta along with the salt. That is called the dry mixing. I'm going to mix it dry. What is the third step? I am going to add some water into it and again start kneading the dough. Once I keep kneading it, what happens? It becomes a chunks or a semi-solid mass. After that, I am going to add a few ml of oil. Why I am adding oil? For leavening the puri. After leavening it, I am going to leave the puri for a few hours, maybe 2 hours or 3 hours. After that, I am going to make a balls out of this puri. Okay, don't take it wrong. I make a lot of pellets of puri pellets. And from that, I am going to roll the puri into a round shaped thing. Then I'll heat the oil in the approximate heat that is required. Then I'll be frying the puri, shooting the puri and taking off. Okay. So this is the normal process. My dear friends, what are the variables that is there present in making a puri? Now, I'm adding a salt. Suppose I'm not mixing properly. What happens? Salt is located in one part of the dough, dry mix, and other part will not have a salt. So what should the critical factor? Mixing is going to be critical. I have to mix it properly. If I'm mixing properly, then salt will be evenly distributed. So one part salt and another part no salt can be avoided. What is the second chance I can do? I can slightly modify the process. I can add the salt in water and make a lukewarm water and add it in the puri. That also I can do it, right? So there is a slight process modification is there. If you're adding it water uniformly, almost uniformly, you can mix the salt in the dough. What is the third one? I'm adding a leavening agent, that is an oil or flare or this is something I'm adding to it and making the puri soft. And I'm leaving the puri for few hours. Suppose if it is a morning time, breakfast time, I don't have a time for leaving the puri dough for 2 or 3 hours in the table. So what I do, I start making the puri, put it into a flatbed, and I just go for frying the puri. Now there is also a variability. If I am going to fry the puri immediately, I am going to likely to get a flat like a papadam. I am leaving it for a long time, likely to get a puffy puri. I'm not a good cook. Maybe you can judge me in that way. And this is a likely, this is the situation that you will be having. Now, I am using the oil. Suppose I have overheated the oil and smoke is very high and I'm going to fry the puri. I'm likely to get a what sort of thing? Very browny puri. Some sort of a brownie I'm going to get. Very fried puri. You have to use the optimum temperature of heating of the oil. So, in making a puri, what are the variabilities you are seeing right from the supplier of atta, whether Annapurna or Pillsbury or XYZ, whatever you are using, that is going to influence the output of the puri characteristic. How thick it is, how easily you can swallow the puri or something, eat the puri. Next one is addition of salt, which is connected with the mixture of the dry mixing of this thing. Then leavening and leaving it for some time. Then making it a puri and shooting it in the oil. Many characters thing. The oil temperature that is there. These are the characteristics that is going to influence the quality of the puri that you are going to eat. Right? So, in the making of puri, these are the variables that is there. These are called as the process parameters. What do you call process parameter? And it is critical for the process. You slightly change, the characteristic is going to change. So that is called as the critical process parameter. Like you come to the situation of a pharmaceutical product. The patient is taking an anti-diabetic drug day in and day out, 365 days, he is going to take the drug and he is taking 2 or 3 times a day. And one batch is going to be one characteristic, second batch taken by a same brand, same company is having a different characteristic. Is it acceptable? So what do? You take up some process validation. For doing a process validation, you need to identify what is the critical process there in the making of that particular drug. Whether mixing, dry mixing is going to be important, like in the case of puri making, that salt addition is influenced by the dry mixing time. Or wet mixing is going to be important, the way you are sprinkling the water in the puri dough and you are going to knead it in such a way that is going to got a uniform wet mass or something like that. In the case of pharmaceutical products, you are going to add a paste or some other PEPK30, a binder is going to be added. You can add the binder at one stretch, or you can add the binder in a thin stream uniformly when the mixer is going to rotate. Those things is going to influence. If you are adding the binder at one stretch, what will happen? The binder will be located at one mass of the dry mass of the powder. So it is not evenly mixed. What happened? And the final thing, one part of your tablet, one part of your batch will have a very less dissolution. Another part is going to be a very lucid or a very easy dissolution. Friability will be very high. Friability will be low in the other part. Dissolution will be low in part. Dissolution will be very high in the other part. Many variations is taking place inside the batch itself. So what is the critical parameter there? Addition of the paste, whether it has to be added in a single stream, upon what time, the paste has to be, what temperature it has to be maintained. All those things will be added into a product characteristic. Not only those things. For example, you are going to dry the product. How do you dry it? First, you go for an air drying. Then you go for a drying itself. What temperature will be drying? How it will be influencing the thing. For example, you are going to make a paracetamol tablet. What are the things that is built into it? The paracetamol is a very common tablet. Making the process or building the process into it a bit complex, because people who have manufactured it will be knowing the criticality. You have to maintain a particular temperature there. I mean, the binder there. The binder is not insufficient. It is going to be very friable. So normally in the olden days, they will be adding gelatin, PEPK30, and also starch paste. Nowadays, they are using only PEPK30 and starch paste are used. Gelatin is out of the context now because of the regulatory stringency over the gelatin. Now, not only those things, in the case of drying of the paracetamol tablet, what you need to maintain an LOD, loss on drying, of 1.5%. The moisture must be 1.5%. Now, you reduce the moisture, the tablet is going to be very friable. You increase the moisture, suppose you increase the moisture and the degradant like 4-aminophenol impurities will be developed at the end of the shelf life or something like that. So you need to compromise between those things, and the chemist must be very vigilant. You must dry at particular temperature. You have to identify and take a product at the right temperature. So this is what we call it as the critical process parameters, my dear friends. So the dry mixing is a critical thing. Addition of paste is going to be critical. Drying is going to be critical. And also what hardness is going to compress the tablet in the compression machine is going to be critical. So this is what we call as the critical process parameter, CPP. Now, we understood the three things. We went through the three things. What is QTPP, quality target product profile, critical to quality attribute, and critical process parameter. Is there any connection between all these three things? Yes or no? There must be a connection. The process parameter, CPP, critical process parameter, is totally linked with the critical quality attribute. That is what we are going to see in the future slides. Okay, sir. Next slide, please. I hope you have understood the concepts, all the three concepts. If you have understood this thing, the next part is going to be relatively easy for me, because that's why we have taken more time in dwelling into those three concepts. Now, in the slide, you can understand, maybe I'm not able to clearly visualize the slide. In the QTPP template, the thing is like mentioned as dosage form. So where do you get this information? We already discussed. You construct a QTPP from a RLD. What is an RLD? Reference listed drug or a reference drug. What is an RLD? Why it is given RLD? Because in US FDA, when you're going to register a drug, you have to identify, do an ANDA, you need to identify a innovator. That innovator is termed as an R&D. So you just try to follow an innovator in terms of all the characteristics like CQA, what you mentioned as say dissolution, everything you need to match it. Even the biostudy you are going to do, you need to match it with the innovator. So this is what you need to do in case of matching this thing. So from the in the QTPP program, what you will be doing, you will be identifying a dosage form. You go for a tablet. Your innovator is a tablet. You do a tablet. You don't do a capsule. To avoid the rejection of the ANDA, you need to follow the many of these things. Like what is the dosage strength you are going to take? Maybe 20 mg you are taking. You take the 20 mg from it. So what is the dissolution you are going to do it? You take the dissolution characteristic, maybe not less than 80% or 80Q or the 75Q, whatever that has been given for this thing, you just take those things. So many of the attributes that is there for the innovator, you try to match those attributes. That's what you're going to do it in the case of the QTPP program part. So next we go for the next slide, please. Next is going to be the critical to quality attribute. So the critical to quality attribute, I'm just rattling the slide, I think. CQA is a physical, chemical, biological, or microbiological property or characteristic that should be within an appropriate limit, range, or distribution to ensure the desired product quality. So my requirement is desired product quality. What is desired product quality means? Already my quality is being given. That is called predetermined specification. My tablet must have this much of dissolution. My tablet must have an assay between 105 to 95. My tablet must pass impurities that is present. My tablet must have a moisture content of this one. Already you specified it. From where you got the specification? From the earlier leader, innovator product, you have got all those details. So from that, you're going to match the thing, match the character. This is called the critical to quality attribute. Okay, sir. Next slide, please. So in the case of my CQA, next slide, please. CQA, I am having a template. I just prepare a template. In this template, I just mentioned what is the appearance. So what is the target? The appearance has to be pale yellow color or a straw yellow color. You just give the thing. Then what is the size of the tablet? What is the dimension it has to go? Then what are the characteristics like what I'll be doing, assay I'll be doing, content uniformity I'll be doing, dissolution I'll be doing. And you give a justification why you're having this particular thing, why you have set a limit like this. This is what it will be added in the case of the CQA. Okay, so next slide, please. Next slide. So I have just mentioned what are the things that will be there in the CQA. We go for the next one, that is the CPP and CQA. What we know, my dear friends, what is CPP? Already you are known, it is a critical process parameter. And CQA is the critical to quality attribute. Already we discussed it. Okay. So now we are going to see the relation between these two things. Well, first is going to be CPP process is blending. So in the case of blending, what is blending? Like in the case of making a puri, atta, you're just going to knead the atta in the dry mix stage along with salt. If you are not blending in the right way, what will happen? The uniformity of salt in the puri is not going to be established. Like in the case of making of a drug, I am going to make a dry mix. I am going to take a starch as an inactive ingredient. I am adding some active ingredient. Maybe I am adding cetirizine, for example. Very less quantity I am going to add. If it is not blended properly, if it is not mixed properly, what will happen? Content uniformity will be issue. Maybe some part of the tablet, the active is more, some part of the tablet, the active is less. So what is the important thing? Content uniformity is one that will be influencing this thing. So what you have to accept, what you have to adjust in this particular thing, you have to increase the blending time or decrease the blending time, optimize the blending time to do this thing. Okay. Next one, milling. What is milling? Milling means to bring the active pharmaceutical ingredient or inactive ingredients into size, reduced size. Okay. So how do you mill the thing? Normally you mill it in a mixie in the case of your house. For churning a chutney, use a mixie for it. The same character in the pharmaceutical arena, we have an OSG, oscillating granulator, or multi-mill or CAD mill, like that is there. Normally, commonly in the pharmaceutical thing, they'll be using multi-mill is used. When you observe the multi-mill, or maybe you observe your mixie, in the mixie jar, statutory warning, while observing, switch off all the thing, don't plug the thing. When you see how the blade is like that, have you ever seen the mixie blade? If you carefully observe, one part of the mixie blade will be blunt or flat. Other part of the mixie blade will be sharp. One thing, it is blunt or it will be flat. Other one is going to be sharp. In the case of multi-mill also, you have a flat area and other one is going to be your knife-like sharp area. Now I am going to put the particles into the multi-mill. If you are running in one direction, and it will be cutting. If it is run in an opposite direction, the particles will be beaten. The action of beating will be something like hammer. If a particle is getting interfere with the back side of the multi-mill, what happened? It's like have an impacting of a hammer there. Suppose they're going in the other way, what happened? Cutting will be taking place. My dear friend, just tell me which will be better. Cutting will be better, you will get a uniform particle, or you will be getting an uniform particle while you are passing through a beating process, hammering process, impact process. Two processes are going. Cutting is there, impact is there. Cutting is in the knife side of the multi-mill. Like in your mixie, there are two sides. One is the sharp edge, other one is the flat edge. In the sharp edge, you put your coconut. What will happen? It is going to cut it in a uniform way. On the other hand, if you're going to put, I don't know whether mixie can rotate in two directions, but I'm taking that analogy in the pharmaceutical arena, scenario. If you're putting it in the other side, the thing is, it is not going to churn it in the right way, or it is not going to give an even particle. So it will be giving in a unsized particle. The same is the case when you're passing through the multi-mill. If you're passing in the knife in the one direction, it will be cutting the particle in a uniform way. If it is passed on the other side, it's going to give an uneven particle based on the impact over the particles. So particles of very fine will be there, particles of bigger size will be there. Maybe such sort of a particle nature will be good for a sustained release formulation. If you are passing the particles through a flat impact is there, that is better. But in the case of an immediate release formulation, which is action is going to take immediately, you need to pass through the nice front of the blade has to be passed through. This is a general thing, but there may be exceptions are there. So like that, milling, what you need to do, so milling will be influenced again in the case of the content uniformity. The particles of uniform size will have a content uniformity will pass. If the particles are not uniform, what happened? Bigger size and smaller size are there, the content uniformity is likely to fail. Well, what are the other things we saw? We went for lubrication. What is lubrication? It's oiling. Again, in a puri scenario, it's a leavening agent we are using. While you are doing this thing, while you are rolling puri, what do you add there? I forgot to tell you that one. While you are rolling puri, what do you do? Can you roll the puri without any other lubricants there? You use the same atta for sprinkling over it to make a puri not getting adhered to the puri stone or puri roller. The same way, when you are going to compress the tablet, the tablets may likely to stick over the punches, upper and lower punches of the compression machine. What do you add? You add some lubricants to it. That lubricants has to have the functionality. If you add more lubricants into it, it is going to influence the formulation negatively. If you add less lubricants, it's also going to influence the formulation negatively. For example, you're going to use some magnesium stearate. Magnesium stearate is a polishing agent for the punches and dies. What is stearate? Stearate is a stearic acid. Stearic acid is having a long chain, lipid chain like. Why you are using this one? Because it's having a long chain, it have a soothing effect to the stainless steel, that is the punches and dies. You add more, what will happen? More than about 1.5% or 2% or 3% you are adding magnesium stearate. It will form a very waxy coating over a drug itself and it will impede in your dissolution that you are going to do. Right? So dissolution will be very much influenced by the lubricant that you are going to add and also the flow of the granule from the hopper into the feed frames. So many of the characters are embedded into it. So critical process parameter, you compare it to the critical to quality attribute. If I am not adding properly the starch paste or the binding agent, dissolution is going to be impeded. If I am not going to use the multimeter in the right direction, right speed, what will happen? The particles will be ununiform. So what will happen? The particle size is very small. Dissolution will increase or decrease. Question for you, my friends. Particle size is less. Dissolution will increase or will decrease? Okay, unfortunately, I need to give the answer. The particle size is small. The surface area is going to increase. So the dissolution is obviously going to be more. Bigger the particle, the dissolution is less. Now you are going to hit the particle or mill the product in the flat side of the multi-mill, then the other thing is going to happen. The dissolution is going to be influenced negatively. If you are cutting it in a even way and you are passing through the right sieve, maybe 0.5 mm sieve or 0.25 mm sieve or 1 mm sieve, what happens? The particles of uniform characters are collected and it will be used in the process. Am I clear? So what happens if you are uniformly collecting, cutting all this thing, the dissolution is going to pass, the content uniformity is going to pass. So this is what is going to happen. You are mixing properly, what happens? Your content uniformity is going to pass. So these are the things that will be influencing. Then I'm going to another one process parameter, compression. When you are compressing the tablet, you need to keep the right hardness. A limit of hardness has to be maintained. How much lower hardness you can go, how much higher hardness you can go. The lower the hardness you are going to fail the friability or you are going to fail for the, friability, or you'll be passing the DT. Of course, you'll be failing for the friability or any other parameters, that is chipping or collaring effect, all these things will be formed. When the case of hardness, when you are keeping it high, what happened? The DT is going to be failed or DT may be, the dissolution may be less. Those things will be there. So you need to optimize the hardness, what Newton you have to keep, what kilograms you need to keep. So the limit has to be maintained in the case of making a compression. So the parameters is closely related to your CQA. The hardness you are going to keep is going to be the critical process parameter. The amount of outcome is going to be measured by the CQA. What CQA are you using? You're using DT as one CQA. You're using a friability as another CQA. Dissolution as other CQA. So 3 CQAs are there in the case of the critical process parameter. Understood, my dear friends? We go for the next one. Next slide, please. So we go for the next very important thing, that is called for the pre-formulation activities. In the pre-formulation activities, when you're going to design, you need to understand you're going to invite a new friend into your flat. What do? You need to understand them or her. Okay, nowadays cohabitation of different sexes very much accepted in the society. Okay, so you need to understand her or the boy. So you understand what you do, what is your hobby. All these things will be coming into this thing. So in the case of pre-formulation, you're going to start with the activity. You need to completely know what is the characteristic of the drug you're going to deal with. What devil is there in it? What are the excipients you're going to use? How it is going to get compatible with that excipients? What process? Hello? Am I audible? Hello.
Yes, sir. Yes, sir. Audible, sir.
Well, so, okay, fine. So we go for the analysis of the reference listed drug product, characteristic for its description. I analyze the RLD also because when I going to take the attributes of the innovator, first I need to understand the innovator. Like that, first I understand the innovator, then I try to copycat that innovator and I know the attributes into my product to reproduce it. So what I do, I just analyze for the description, see the description, coated or uncoated, type of coating, dimension, weight, physical appearance, disintegration test, assay, impurities, everything I just identify. Okay, dissolution, everything. Now, in the case of pre-formulation, evaluation of the drug substance properties like physical properties will be seeing. Already we saw, my friend, physical properties, what you will be doing, you will be checking for the solubility. Not only solubility, you will be checking whether the dimension, what particle size I am going to take. I am going to take an amorphous particle, I am going to take a crystalline particle or something like that. If I am going to take an amorphous particle, what is the size, particle size I am going to take? I am going to take a particle size of 10 nanometer particle size, or I am going to take a 2 nanometer particle size or something. The problem is you need to have a practical approach because the lower the particle size, the solubility is high. But the lower the particle size, you are going to take a 2 nanometer particle size. What happens? The static activity will be very high. If you are going to use a container, it will be clinging to the container and won't be coming out exactly out into the vessel. So a lot of problems are associated. When you're going to go for one extreme, you need to also compromise on the other thing. So you need to optimize between this thing, particle size and dispensability of that particle into a during the process area. Okay, next one is going for your physical properties we saw, then description. What description we need to have? For example, you're going to design a product for a pediatric drug or a geriatric drug. Geriatric is for the old people. Can I design a formulation of 6 mm tablet, very small tablet, or even less than 6 mm tablet? You can't. So you need to go for a slightly higher size tablet, maybe 10 mm or 12 mm. Those tablets will be taken for geriatric consideration. And dispensability of the tablet, what containers I'll be taking, I need to for a child-proof cap. Child can't easily open this cap. So you need to press the cap and roll it for opening those things. A child can't understand that mechanism of opening the thing. Those small details in the container packing system is also going into our development of a product. Not only the product, you have to see also the container closure system into development of this thing. Next, we go for the solid state form, XRD. So X-ray diffraction you will be taking, you will be identifying what is the crystalline form, whether it is an amorphous, whether it is a crystalline, if it is a crystalline, whether it is a form 1 or form 2, all those things will be taken to it because you are going into a thick forest of patents. So when you are going for the patents, you need to bypass the patents and go. And if there is any exclusivity or patents are there for the area innovative product, you need to take a very cautious approach. So these details will be required. Then solubility in functions of pH. So solubility in function of pH is normally we call the age-old concept called BCS concept. BCS bioclassification system. We have classes of drug, 4 classes of drug are generally classified. Now it is not much accepted. There are other classes are also the concepts are there. But it's old and it's accepted to an extent. What is BCS class 1? The drug is classified into 4 classes. Class 1 is going to be highly soluble, highly permeable. That means highly soluble means hydrophilic. Highly permeable means lipophilic. So lipophilic and hydrophilic are there. Hydrophilic means soluble in water. Lipophilic means soluble in lipid agents. So product which is soluble in water is going to have a peripheral activity. Peripheral activity means what? In your body, limbs, all the stomach, those areas it is going to act. A lipophilic drug is one which is having a solubility in oil. So it can pass through the blood-brain barrier. For those drugs which are going to have a central activity. What is central activity? The drug that is going to act in the brain or CNS or other areas related to the central activity must be having a log P value high. Log P value means solubility. The higher the log P value, the higher the solubility of the drug in the lipophilic agent, then higher the permeability of the drug into the bloodstream through and go into the cross the blood-brain barrier. Well, Now, how the log P will be increasing? If the drug is having a long chain, you'll have a long chemical, CH310, it is going to have a long log P, or the lipophilic characteristic is going to be very high. You have read in your +1 or +2 chemistry, if my memory is going correct, you have read a concept called Anderson-Hasselbalch equation. Yes or no, my friends? Maybe your children is doing for a NEET preparation, they are doing this thing. In what is that concept there? What is Anderson-Hasselbalch equation? Degree of concentration of ionized by unionized. My question to you is whether the drug is an ionized form. In ionized form, it will be absorbed fast into the central thing, whether it will be absorbed fast or it will be absorbed less. My question is this thing. And unfortunate thing is I need to give the answer because it's going to be an online lecture. I can't see your face also. Well, the drug is in an ionized form. Unionized form, it is intact form. It will be absorbed more from the stomach because the gastric lining, it can be absorbed very fast. If the drug is in an ionized form, means broken down into ions, it won't be easily absorbed. Okay, in the stomach stage itself, it won't be absorbed. So you can compare the concepts of omeprazole and many of these things. Maybe you can ask a question. Omeprazole is a drug which is going to act in the stomach, and it has to reduce the ulcer nature, but it has to be absorbed into the bloodstream to have an action in the stomach itself. Have you ever thought about it as a pharmacist? But in the case of an antacid, it's going to be different. You churn it or you chew it, and it's going to have an action, reducing or neutralizing the acid, excess of acid that is present in your stomach. But the case of omeprazole is not the case. Cimetidine is not the case. What happened? It has to be absorbed into your bloodstream to have an action, the same area of your stomach where from it was absorbed. So coming back to the thing, we came for the BCS class 1. BCS class 1 stands for the lipophilic and hydrophilic, both is there. What is class 2 standing for? The drug which is not soluble in water. It is not hydrophilic. It is only lipophilic. That means it will be only absorbed, lipid loving, it will be absorbed by the oil. It will be going into the solvents. Class 3, hydrophilic, but not lipophilic. Class 4, both the things are absent. It is not lipophilic, it is not hydrophilic. Nowadays, the DCGA is stipulating if you are going for a class 2 drug and class 4 drug, you have to do the bioequivalent. In the case of class 3 drug, you can have a limited justification. With that, you can avoid the bioequivalence. Those things are being there. Regulators are more stringent with this particular classes, with this thing. So you need to identify what class of drug you are dealing with. So what sort of a culprit is your drug, how you are going to deal with this drug. If it is going to be very lipophilic in nature, you need to add an excipient which is going to be hydrophilic in nature. Then only it will get slightly dissolved. So you need to formulate in such a way. So you need to add more of lactose into it. And again, while you're adding lactose, you need to take into consideration whether a primary group or secondary amine group is present in your drug. If a primary or secondary amine group is present, it is going to have a age-old reaction called Maillard reaction. What is Maillard reaction? It will make your drug into brown color when you are heating or giving some energy into it. So this brown color can go into a dark brown color. If the tablet is uncoated or is slightly white coated or something, the reaction is going to be visible to the patient. The patient will be thinking the drug is deteriorated, but actually it is the process that is reaction that is going on. So you need to take those consideration into this thing. Okay, coming back to our discussion. So solubility in pH based on the BCS class. Next is hygroscopicity. Hygroscopicity is a very tandem thing, very tricky thing. If the drug is going to be very hygroscopic, it is very difficult for us to process. So you need to add some absorbing, for example, dibasic calcium phosphate or something. You need to embed the drug in some other substance, inactives like that, and try to do this thing, or take the drug in solvent and try to disperse it into the inactive ingredient. So the process you need to consider in such a way. You need to have an area that is going to be a lesser RH, that is relative humidity, less temperature. All this exposure of the drug has to be less. A lot of issues will be there in case of hygroscopic drug. So in the pre-formulation, you need to identify based on this thing, you will be identifying what are the processes you are going to take. Next is going to be your density. Density, we have a lot of densities there. Bulk density is there, tab density is there. Then we also have a true density. True density is the void that is present between the air and the water that you add. So you identify the density of the product. If the product is going to be a less density, it is going to be very fluffy. For example, you have a colloidal silicon dioxide is going to be few grams is going to be very, very high. That means it's very fluffy natured product. I mean, inactive ingredient. So the handling of the drug is going to be, if it is going to be very fluffy, you need to identify what way, what process method you are going to do it. If the drug is very fluffy, you can't go for a direct compression mixture. Then you need to go for some other granulation method or a roll compaction method. So such of the things you need to identify. Then is your angle of repose. What is angle of repose? In your, again, my dear friends, in your third year or second year B.Pharm, if my memory goes, I think I did it in third year B.Pharm. So you put a funnel in the burette, stand, and you pour something, a granule into it. The granules will be coming and landing in the form of a cone in a paper, and you just put a circumference around the base of the cone. Then you take the height of the cone. Then you calculate the theta, that is the angle of repose. The theta, if is going to be high, the granule is going to be free flowing or not free flowing. Can anyone say? Well, if the theta angle is less, the flow is good. Okay. So iron pellets or iron granules you're going to put into the thing, and you're going to check the angle of repose. The angle of repose is going to be less. You are going to take some sand, it is going to be slightly more. You're going to take some other aerosol or colloidal silicon dioxide, then the angle is going to be very high. So based on it, you can do it. There is also a very good chance for you, maybe students or people in the teaching faculty can explore. We don't have in the industry a thing called reposimeter. Earlier, they used to have, maybe 25 years back, there used to be reposimeter. With the reposimeter, we used to identify the granules. We put the granule, we don't measure the theta and all. In the board itself, we just put a red line, green, yellow, or something like this already there. We pour the granule through the funnel, where it is going and which area it is going to go and sit. Based on it, we'll identify the granule is good. The compressibility will be good. Based on it, we'll identify. In the scope of you, maybe you identify some 200 granule, make a reposimeter, give it ready-made to the industry. Good scope is there. Well, it's going out of the topic scope of the discussion. Next is melting point. What is melting point? You know, a very pure substance is going to have a very sharp melting point. If it's an impure, it's going to have a melting range. Then we go for the next one is eutectic mixture. So eutectic mixture is lowering of the melting point. Normally menthol, peppermint, all these things. When you're adding to other substance, what happens? Many of the substances will have a reduced melting point. So when you're adding something like that, formulations of that nature, when you're considering, you need to add more of an inactive ingredient where the heat of formulation is absorbed and it is dissipated by the inactive ingredient. Then crystalline nature, already we discussed the crystalline nature. So crystals are less absorbed in the dissolution. You have to go for an amorphous, that is crystal, non-crystalline nature, which are brought into very small particle size without any shape. It is called amorphous. So thermal analysis. Thermal analysis, what do you do in the pre-formulation? We have differential scanning calorimetry, thermogravimetry. Methods are there. What do you do in the differential scanning calorimetry? There is a small aluminum pan wherein you will be keeping the drug. And you take you give a temperature for the drug through the pan, an uniform temperature. Different menus are there. You can give a very high temperature or low temperature, slowly incremental temperature. You see the nature of the drug. At what time the drug is melting, you just see. Now, in that you add some inactive ingredient, you can again give the temperature. You see if there is any interaction between the drug and the thing. There may be some glass transition temperature will be there, or the melting point will be shifting. Glass transition temperature will be changing, which indicate the drug is not compatible with the inactive ingredient you are going to add. So, next coming thing is drug compatibility, wherein you can use this method. Thermogravimetry method is for maintaining or identifying how much of water is there, how much of water is bound, how much of water is left from this thing. Those aspects we'll be doing. So we don't want to detail most of this thing. We'll go to the other thing. Next is stress analysis. What is stress analysis? How compatible are you? How manageable are you? Maybe I'm going to give a kick to you. You regain your composition. You still you keep calm. That's good. I'm going to give a big blow for you. Still, you regain your composition and don't retaliate over me. Then you're even more good, like a Gandhi, non-violent action you're having. But reality is that case. I take the drug, active ingredient or the API, active pharmaceutical ingredient, I am going to give a stress to that particular guy. What I do, I give some acid, N/10 hydrochloric acid, I just pass 0.1 in hydrochloric acid, 0.1 in sodium hydroxide, or 1 in hydrochloric acid, 1 in sodium hydroxide, potassium permanganate, hydrogen peroxide, heat I'll be giving sunlight to it, I'll be giving excess of thermal to it, light ray to it. I'll be exposing drug for a few hours. I'll just waiting for 20 to 30% of the degradation of the drug. I just inject in the HPLC or any other suitable LCMS or something. I just see how much of the drug is getting degraded. That is what I call it as a thermal, the stress study. In the stress study, what I'm going to identify, what agent is going to degrade, whether moisture is going to degrade my drug, hydrolysis is taking place, or presence of oxygen. Oxidation is going to be a degradation pathway for my drug. This I'll be identifying using the stress study I'm going to take up. I'll be checking the acid compatibility. It's an acid-based degradation or base catalyzed reaction degradation is taking place. Those things, the kinetics of the degradation, I'll be identifying by passing the drug through the stress study. So I'm going to get to know my drug very much. That is what earlier I said. When you're going to take up this thing, you need to identify what devil is there in the drug. That will be identified through your pre-formulation. So those are the characteristics we'll be doing with the pre-formulation. Not only those things, I'll be identifying what is the LOD and moisture content. My dear friend, what is the difference between LOD and moisture? LOD is loss on drying. When you are going to give a heat, the amount of moisture that is going to be left out, water that is going to be left out from the drug is going to be calculated by the LOD. But in the case of moisture content, what you will be doing, you have to do the titration. So there will be bound and unbound moisture in the crystal, API crystal or KPI. So that will be estimated by means of the moisture content. In the case of LOD, what is unbound is going to be left out. There will be some amount of bound moisture. Okay, so moisture content will give a more result when compared to the LOD. So you need to identify those aspects also. What temperature you're going to see this thing. Next one is last but the least, partition coefficient. What do you see in the partition coefficient? Already we had a long discussion about the partition coefficient. That is the log P value. So octanol and water, you're going to add a drug into a beaker of octanol equal quantity of water. So when you're adding octanol water, octanol will be floating at the top and water will be heavy and it will be coming down. Suppose I am taking chloroform water, where chloroform will be there? Bottom or in the top? Friends, can you interpret yourself? Be honest and tell to yourself. Well, chloroform will be down, water will be up. Now, in the case of octanol water, that is commonly taken to identify the partition coefficient of a drug, to identify whether the drug is soluble in oil or in water. This characteristic I need to identify to know whether what characteristic, what BCS category the drug is falling into. So we identify those things also. Then we do the excipient compatibility study. In the excipient compatibility study, already we told or we discussed, I'll be adding a drug and I'll be adding an inactive ingredient. I'll be checking the compatibility of the drug with that inactive ingredient. If it is not compatible, I try to avoid it in the formulation. Then particle size distribution study. What is particle size distribution? We already discussed, the lower the particle size, the better the solubility. If the solubility is good, dissolution is going to be good. So particle size distribution I will be taking in the form of bell curve. If it is going to be a bell curve, the bell curve 90% of the thing is going to be in the center of the bell. There will be some extremes. We call it as a D90. Some cases D70 will be there. That is the distribution 90% is of a particle 10 micron or 10 nanometers particle. Whatever the characteristic you want to give for the drug, you need to characterize the particle size. Particle size is normally characterized using a Malvern or other particle analyzers are also there. Mechanical particle analysis are wet method is there, dry methods are there, wherein you will be using the same. Now, next, so how much time we have, sir? What is the time now? We have we have only almost we have gone through 1 hour, so we have around 25 minutes, right? So thinking about the situation, I will try to skip few slides. Skip means I don't going to skip this. Go for the next slide, please. Design of experiment. Design of experiment already. My student, my friend Josna has already given in the forenoon. That was before lunchtime. So you would actively listen to her discussion. So I try to skip those things. But however, when you're going to take this thing, we need to have the thing into you. I need to tell you a basic of formulation development into you. Now, I am going to take design a drug, for example, cetirizine tablet. I don't know what is the mg of active ingredient cetirizine. I think it is to 10 mg. I think so. For example, 10 mg. I think cetirizine is 10 mg. I am going to buy a RLD from the market. I weigh the tablet. The tablet is weighing 100 milligram. Now I just take a pen, paper. In the desktop, I'm going to do the designing of the experiment. First, start with this thing. What I'm going to do, I just write cetirizine is 10 mg. Then I'm going to take another one called PDR, physician drug reference, desk reference. Nowadays, that is not required. You have RX list in the internet. You can open this thing, identify what is the inactive ingredient that is present in the RX drug list or drug banks or something like that. From the internet, you can find, identify what are the inactive ingredient that is present in cetirizine. In the paper, you just write down. First, you have written cetirizine 10 milligram. Next one, you are going to write lactose, for example. It is mentioned lactose. You just write. You don't know what is the quantity of lactose. Next one is what you're going to write is starch. You don't know what is the quantity of starch. Next one, you are going to write cross caramelose sodium. Write it down as CCS. And next one, you're going to write it as magnesium stearate. So magnesium stearate, you just write it down. And finally, you know the weight of the tablet is going to be 100 milligram. You purchase a drug as 100 milligram. You know the weight of the tablet. So you write at the last column 100 milligram. First cetirizine 10 milligram, you know. But the rest of the thing you don't know. So in the desk paper way, how you are going to do it? You have read in your Leon Lackman or some other books, how much of CCS, cross caramelose sodium, you have to use in a formulation? Yes or no? Yes. They say 3%. So 3% of 100 milligram, just write 3 milligram, right? I'm poor in mathematics. So you write 3 milligram. Then you are going to add some magnesium stearate. How much of magnesium stearate you normally add? If I am a formulator, I'll prefer 1% of magnesium stearate. I don't go beyond 1%. Maybe in worst case, I go for 1.5%. So I take 1% of magnesium stearate. I'll add 1 mg. So what are left now? Lactose is left, starch is left, maybe a binder I didn't forget to mention. PAPK30 is a binder I'm going to add. So PAPK30, how much I'll be using? I'll be using 2% or 1.5% or 3%. I take the worst case 3%. For my 100 milligram of total tablet, 3% is going to be 3 milligram. Again, I am poor in mathematics, correct me if possible. So 3 milligram I'm putting the table. So almost I have solved all the things. I know what is the quantity of cetirizine. I know what is the quantity of SSG. SSG is a disintegrating agent. How much I'll be adding? Maybe 2% or 3%. And for 100 milligram, how much of 2%? You just write down. Magnesium stearate, you know, you write it down. Now the binder, you're going to use it. That also, you know, you're going to use 2%, you write it down. What is left off now? Lactose is left off. Maybe you are using MCC. Sorry, you will use instead of starch, I use MCC because you get a good granule when you are using a cotton, microcrystalline cellulose. Normally, MCC, if I am using more, that is going to be a headache for my formulation. Why it is going to be a headache? Because during the dissolution, MCC is going to cover your drug and is going to form a heap and it's not going to allow your drug to go for a good dissolution. That is a big headache. Every formulator faces a headache. So what they face, they keep the MCC normally 10% or 15% or the maximum 20%. After that, they don't try to increase more than that. Normally, when I'm going to be a formulator, I go for 10%. Suppose I'm not going to use the right MCC, what will happen? During the punching effect, the cellulose, microcrystalline is nothing but cellulose. It's going to give a cushion to the tablet. So the punching, there will be a problem. The wear and tear of punches will be very more. You have to replace the punches or the formulation itself will have some roughness will be there. So to smoothen it, you will add a MCC into it. Normally, we add for a 10% or 12%. We assume I take a formulator, I take 10% of MCC. My dear friends, again, I am not good in mathematics. 10% of 100 milligram, I think it is 10 milligram. Okay. So you put 10 milligram there. Now you have listed out everything. What you have listed out, cetirizine is 10 milligram. Lactose, I don't know. MCC, you put 10 milligram. Binder is PVK30, you add 2 milligram, 2%. You add a SSG again 2 milligram. Then you add magnesium citrate 1 milligram. The final weight is 100 milligram. You need not be a rocket scientist to identify what is the weight of lactose. You add all these things, subtracted from 100, you know how much of quantity of starch you're going to use it. Now, tell me from your heart, are you not a scientist now? Yes or no? Sitting in the desk, using few advancement in the internet, all this thing, you are able to jot down what is this thing, what is the list of the thing, and you found out a formulation. Now, it is not exactly correct what an innovator has used. Maybe the innovator has used 15% of MCC, you use 10% of MCC. Maybe the starch what you are using is, the binder what you are using less when compared to the innovator. Now, what all you are going to do is design of experiment is to identify, fine-tune this formula. This is what you are going to do it. My magnesium citrate is 1%. Suppose my magnesium citrate is 1.5%. I design an experiment with 1.5% of magnesium citrate, 1% of magnesium citrate, 2% of magnesium citrate. I check the functionality of that particular excipient, whether it is okay working for me. Likely in the case of a binder, I'll be adding 2% of PUPK acid, that is the binder. I add 3%, I add 1%. Check which is going to be good for me. The binding is okay. Binding in terms of the dissolution is okay, because binder influences the dissolution. We already see CPP is correlated with the CQA. So we check the correlation between this thing in terms of the design of experiment. If I'm having a lower end, middle end, higher end, this is called as the factorial design. My addition of binder is going to be 2% that I have determined in the desk way by sitting in the paper. I make a design of 1% of the binder, and I also make a design of 3% of the binder. What happens? This is called the factorial design. There is other one called level of design. What is level of design? Different designs I'll be incorporating. Maybe I combine the addition of binder along with the addition of MCC, level of MCC, two designs I'm going to combine together. That is called the level of design. That is called 2-level design, 3 factorial design. So if that is the case, you multiply 2 by 3, how much of experiments you will be getting? You will be getting 6 design of experiments you will be getting. So you construct the design of experiment based on this thing. Okay, so already it is covered, so I don't want to waste the time. We go for the next slide, please. And we are also short of time. Now comes the thing, dependent variable and independent variable. What is this thing? Statistics are very allergic, sir. Maths is very allergic. Statistics is equally allergic. Though it is slightly colorful, it's also an allergic topic, something maybe people, but I felt statistics is a very, very important thing we need to take in our daily life. And during a professional life, the statistics is going to be influencing. I also regret sometimes why I'm not gone a full-time statistics course in this case. Now, coming back to the topic. Dependent variable, what is dependent variable? What is an independent variable? The sun rises in the east and sets in the west, whether it's a dependent or independent. You get up at 5:50 in the morning during a summer month. You say, you set an alarm or clock or something like that. Sun come, rise in the east. Will the sun listen to you? It doesn't listen to you. And you set a time until it's 5:58 in the evening. And now the time you can go down. You can go to the horizon and take a rest. Can you tell to the sun? Can you dictate to a sun? You can't dictate anything. Can you tell a time? Time. You have to run fast. You have to run slow. Of course, time runs fast and slow depending upon our fate. And the fate is negative, it runs slow. If fate is good or something, it runs optimally fast or something like that. But it's an analogy only. It is only in situation wise. But really, you tell a time, you run fast. You run slow. It can't happen, right? So how do you categorize it? It is an independent variable. It doesn't listen to you. You or me, it doesn't listen. You tell the weather, you have to be very cold today, you have to be very warm today. Will it listen? These are all independent variables. Natural things are all independent variables. What are dependent variables? Dependent variables are what we set, what we give to the thing. That is called dependent. Those are called as the dependent variable. Normally, in the thing, when you are going to draw a graph, you put a dependent variable in the x-axis. Where does the x-axis come? It is the horizontal one, is the x-axis. I mean, independent variable will be in the x-axis, and the dependent variable in the y-axis. This is what is given. Time is an independent variable. You can't control time. They call it as a third dimension. Time is a third dimension, they call it. Still, we take it in the statistics in the x-axis. So you can see some of the examples there. What is the dependent variable, independent variables, everything. Again, I skip this slide also because considering the time, we have very less time availability. We skip this thing. We go for the next one. Well, next. So design of experiments at factorial level. I told you already, factorial means the lower level, median level, higher level. These things will be combined to called as a factorial level. You can have n number of factorial. You can have 4 factorial, 5 factorial, 3 factorial, 2 factorial. Many factorial designs you can make. Well, next slide, please. So in the thing, next slide, please. Oh, EL1. Yeah, okay, we'll go with this thing. So here you can see factor A, B, C. One is the mixing time is changed. B is magnesium stearate concentration. C, addition of lubricant, talc. So concentration. 3 different factors are taken. 3 different levels are there. Factorial is going to be 3 factors are there. 3 factors means low, high, medium, or the 3 factors that is there. 2 level. I just 2 level design is there. 3 factors are there. 3 factors we are taking is mixing time, magnesium stearate concentration, talc concentration that contributes the factor, and the level is going to be a lower level and higher level. So you can identify number of experiments. You can construct a number of experiments from this one. These things. Okay, next one. Next one is confounding of design of experiment. Again, I don't want to go in depth because some of the attributes what you're going to measure in the design of experiment, you can omit it and you can take some what is blatantly seen. So confounding is applied in the design of experiment. Next slide, please. Next one is excipient to drug incompatibility. We already saw you mix one drug with other API in one excipient with a drug in a 1:1 combination or 1:2, whatever the case you just you take it to an HPLC analyze. Any degradation is taking place after some time, or you can also pass it through the differential scanning calorimetry, that is thermal analysis. You identify the compatibility of the substances. Next slide, please. And even next to that. Next one is next slide, please. Yeah, stress study. You can see the typical stress study that has been given there. You pass it through an acid, you pass it through an alkali, pass it through potassium permanganate, you pass it through hydrogen peroxide. Sometimes you pass it through light of different lux value. Just pass. Identify how much of degradation is taking place over some time. Normally 20 to 30% degradation is acceptable. Because if it is degrading very 75%, then what happens? There may be a degradant for the degradant again. So that becomes very complex. So normally a mild degradation of 20 to 30% is taken for stress study analysis. So we go for the next one, please. I think okay, next slide. Okay, excipient grade and vendor selection. We discussed Pillsbury atta with Annapurna atta and XYZ ABC atta, all those things. So whatever excipient you're going to say, whether it is regulatory accepted, those things you need to characterize the vendor. You identify, take samples, multiple samples from the vendor, you characterize it, then use it for your activity. Next slide, please. Flow function coefficient, how the flow is there. Maybe the powder has to pass through the hopper and it should not give a rat holing or any other feature. So easy flow of granules has to be established. So that slide represents the flow function coefficient in terms of the hardness. So flow function is a critical parameter. Hardness is going to be a critical to quality attribute. The comparison is given as an example. Next one, sir. Next slide, please. So formulation development, we are going for an important thing. We had a discussion about the formulation development. How do you do those formulation development? So identify the lactose MCC ratio and the excipient disintegrator level. So whether you're going to add magnesium stearate in the extra, I mean the inactive ingredients in the extra granular, Some cases you will be adding intragranular. So you need to identify those things. Then what method you're going to do in dissolution method is as per the abbreviated new drug, or you're going to develop an in-house dissolution method. US FDA database has a dissolution method which you're going to follow. So you can identify what sort of blend uniformity, how you're going to monitor it, whether you're going to do the monitoring of blend uniformity by HPLC or use an NIR, near infrared. Now the US is getting official with the planning to go official with the NIR. NIR is the Raman spectroscopy which has been used, and that has been widely. It is a very versatile one, but it's least accepted. Maybe it's because it's developed by an Indian scientist or something. But it's going to be a very versatile tool in future. So how much of granulation you're going to do? When you're going to run in an RMG, that is called rapid mixer granulator, how much of torque value or how much of amperage you're going to give? Amperage is a measure of how much of kneading has taken place. If it is kneading, is initially is going to take more time because the blade has to churn through. Like you're going to press the atta, initially it is going to be very cumbersome. After some time, it has formed a uniform dough. The mass is going to be uniform and the amount of mixing is going to take less energy from you. Same is the case in the machine. When you're going to knead the dough, initially it is going to take more time. Mix the drug and the excipient is going to take more energy. That will be measured in terms of amperage. Later stage, it is going to less, or based on it, you have to identify the end point. So those are the things that will be coming into the design of experiment and your formulation development. Then you go for the next slide, please. Then you go for the process development. You identify what sort of granulation you're going to do. You're going to do a water granulation, or you're going to do an IPA granulation, or direct compression, or a roll compaction. So all these things has to be critically identified based on your knowledge and also by an experience and from the innovators' skills, everything you want to add in this particular product process development. Next one. So finally, you develop the qualitative and quantitative formula. We have to identify what is the unit formula for a particular dosage unit. You need to identify what is the qualitative nature of the excipients. We will be submitting for the registration, all this thing. Next one. Next slide, please. So scale up, normally scale up process we take in the lab scale. After that, we take a pilot scale. After that, we take PO batch, process optimization batch. So those process optimization batch, then we go for the exhibit batch. In the case of exhibit batch, the batch size must be minimum 100,000 units. Whether it's going by tablet or a capsule or a liquid oral substance units, you have to make 100,000 units minimum for an exhibit batch. 3 exhibit batches normally taken. That exhibit batch will be loading in the stability chamber. The stability is pakka after 6 months and 1 year, accelerated on the real time. Then you go for filing the particular thing. After that, after reviewing the document, you can go for the process validation batch. Again, process validation batches for 3 batches you need to take. The batch size must be minimum 100,000 units. Again, we'll be loading in the stability and you will be filing in the regulatory thing. Everything is okay. You can trigger a inspection of your facility. This is how it goes. After that, you can go for a commercial manufacturing of the product. Next slide, please. Risk analysis you need to do. Already we took critical process parameter against the critical to quality attribute. My particle size is going to be critical for dissolution. Yes. My particle size is critical for the content uniformity. Yes. If that is the case, you have to put red square in this thing. My particle size has no influence on the assay of the product. Whether the particle size is this big or small, the assay is not going to influence. So it is having a least influence. You can put on green or yellow or something like. So you need to characterize each and everything with the particular CQA. That is what the risk evaluation is there. You can also map the vendor details also into this risk evaluation. A process detail also you can put it in the risk evaluation. A detailed risk evaluation has to be given and a summary of this thing has to be dwelled and it has to be prepared. The risk report has to be prepared for your formulation that you have developed. Next slide, please. Next slide, please. So you need to give a justification. Then you have to give a risk assessment. Next slide, please. And you need to give a risk categorization, whether it's high, medium, or low. Next slide, please. So, drug substance and flow. Yeah, this slide has to be come earlier. By mistake, it has come here. Next slide. So, you will be using a statistical tool, results and interpretation you need to. You can't just give the result or something like that. You need to have an ANOVA analysis. or a hypothetical analysis, null thing has to be done, null hypothesis you can do. You can use a student t-test, which analysis you're going to use, which part of this experiment has to be well defined. So that is what you have to do. Maybe there are softwares like SPSS, and you have Minitab and many softwares are there. You can very well use those things. Stat pads are there. You can use it for interpreting your results of analysis, and you need to make a detailed interpretation based on it with giving a conclusion of it. Next one. So you have to do a dissolution method development has to be done. You have to identify the container closure. Analytical method development has to be done. Compatibility studies has to be done. Product life cycle management and continuous improvement has to be there in the phase. This will be a system building approach with the product also. Next one. Next slide. Yeah. So with this, we have an end of this thing. Thank you very much for bearing with my dry topic. I hope I had drilled in some amount of this thing into you, but I'm still not sure how much has gone into you or something. Well, anything to discuss, sir?
Thank you very much, sir, for your nice presentation.
Thank you.
Thank you, sir. After 2001, I listened to your class again in 2021, sir. After 20 years, I listened to your class.
Years.
After 20 years, sir, same day, I listened to your class, sir. Thank you very much, sir. I feel very happy to listen to your class today.
But I have become old or young? Correct.
Thank you, sir. Thank you.
Okay.
This lecture, we are very happy to hear such type of nice presentation from sir. As a token of appreciation, the Adamachara College Pharmacy sharing e-certificate, sir. After completion of the STTP, we will send you this certificate to your mail, sir. So please accept our certificate.
Thank you, sir. Thank you.
Thank you so much, sir. Thank you so much, sir. With this, we are intending concluding today's
Thank you.
conference. Tomorrow at 10 a.m. we'll start the 6th day session. Thank you, sir. Thank you so much, sir.